Welcome everyone to the third annual Invasive Shothole Borers Fusarium Dieback Symposium. I'm Randall Oliver. I'm the statewide communications coordinator on invasive shothole borers with UC Agriculture and Natural Resources. I am joined today by an incredible lineup of subject experts. And rather than introducing them all here and their topics, I'll ask you to refer to the meeting agenda. And I believe my colleague Julie Clark is putting that link into the chat. So refer to that if you're interested in seeing the order of speakers and what they're talking about. If I achieve nothing else today, I do want to familiarize you with www.ishb.org website. This is the central repository for all things connected to this pest disease complex. On the homepage of ishb.org, you will find an overview plus an introductory video, a link to our free 1-hour online course, and that also has CEUs from both DPR and WCISA. There's an online detection and management tool, upcoming training and events, what's new area, and a sign up for emails. And then if you dive deeper into the website, go to the different tabs, you'll find a wealth of information, including updated mapping from our ArcGIS, which shows the distribution of the pest. There's wide variety of handouts, and that includes probably the definitive review of this pest, which is the UC Integrated Pest Management Pest Note. You'll find videos, news archive, photo archive, and much more, including a number of very helpful links to other resources. A few other thoughts that I hope you'll take away from today. First of all, this problem is not diminishing. It's not going away. Some people think that because we've heard less about outbreaks in Southern California, that it's gone, we're done with this. It may have been successfully managed in some locations, and that's really cut down on the infestations. But when we stop looking for it, it pops back up, sometimes in the same location, sometimes in new places. We saw that in Anaheim after the pandemic, and more recently in Southern Orange County at Ladera Ranch. And now we found it in Northern California. But I'll leave that discussion about the distribution for our next speaker. Secondly, this problem probably can't be eradicated. It's just too widespread, too well established, but it can be managed. So anybody that tells you there's nothing you can do about this, that's just false. One of our speakers is going to be talking about the various management approaches that can be used for this. However, doing nothing is not an option. This is a serious problem, huge consequences for our state, and it needs to be managed. It cannot be ignored. And one key component of that is continuous monitoring. As I said before, if you're not looking for it, you're not going to find it. So let's take a quick look at the biology of the beetles and the fungal complex. The term invasive shothole borers is a common name. It refers to several species of ambrosia beetles. They're native to Southeast Asia. These beetles are very small, about the size of a sesame seed, or if you look at this photo over here, it's about the size of Lincoln's nose. The species include the polyphagous shothole borer and the Kuroshio shothole borer. Both of those are present in California, as well as the T shothole borer, which is a more benign pest, and that is not in California. The Mediterranean oak borer, which you may have heard about, is a similar sized ambrosia beetle that has been found in 4 Northern California counties. That will not be the focus of today's discussion. We have heard some reports that maybe people have found one of these pests in the area like around Folsom. We're thinking that that's probably Mediterranean oak borer because that is present up in the Sacramento region. I mentioned a moment ago that these are ambrosia beetles. So basically what that means is that their food source is not wood. They're not out there eating the wood, but rather the symbiotic fungi that they bring with them when they tunnel into their host trees. That fungus is the main thing that kills the trees over time. And that's why we refer to this tree-killing combination as invasive shothole borers, Fusarium dieback complex, because it's not just the beetles, not just the fungus, it's the combination of the two of them. And Fusarium refers to the main type of symbiotic fungi that they carry, although there are also other types of fungi as well. So the beetle's life cycle begins with a female beetle attacking a healthy host tree. She tunnels into the tree and creates what are called galleries, where she lays up to 57 eggs. Most of those are fertilized and will become females. A very small number are unfertilized and they become males. And then the fungus is introduced into the gallery wall through that tunneling activity. The developing larvae feed on the fungus in the gallery, and when they are mature, they mate with their siblings and or with their mother. Adult females leave the gallery, and as long as the host tree remains viable, they form new galleries in that same host tree. Meanwhile, that fungus continues to colonize the wood beyond the gallery wall, and how quickly that takes place depends on a number of factors, including the host species and climate factors. Over time, the fungus causes branch dieback and tree mortality, and as the tree dies, the adult females then will leave that host tree and find a new one. Then they start that whole process over again. Like most other invasive species, shothole borers have the ability to multiply extremely rapidly. Assuming just 5 females from each brood survive, you could have close to 100,000 beetles in that tree in just 7 generations. So it takes somewhere between 25 and 50 days for the larva to grow into the pupae and then adults. And that's under ideal temperature conditions. So models based on the effect of temperature in their development suggest that ISHBs can have 5 to 11 generations per year in Southern California. So this can obviously happen very quickly. 247 species of trees are attacked. 78 species of trees can support ISHB reproduction, and 17 different types of tree hosts can be killed. You can find all that information in the UCIPM pest note. And again, there's a link in the chat. You can also go to ISHB.org, look under resources to find that. There are some particular favorites. Box elder has been described as the candy or the ice cream for this pest. They absolutely love it and they can kill it very quickly. They also like trees that develop in more riparian types of environments, sycamores, cottonwoods, willows. Those are all among its favorite targets. So what's the potential impact of ISHB-FD? In severely infested trees, the beetles and their associated fungi can kill trees. They can cause branch dieback, tree death. Trees with heavily infested branches can be especially hazardous because the combined damage of the fungal disease and the beetles' tunneling activity weakens the wood, causing limbs to break and fall. Sometimes they fall on cars, sometimes on people. And dead and dying trees also provide fuel for wildfires. So obviously not something we want in California. 2017 study found that 1/3 of Southern California's 71 million urban trees are susceptible. And if just half of those die, the replacement cost would be nearly $16 billion. That does not include lost ecosystem services or lost aesthetic value. And those factors were looked at on a smaller analysis done 2 years ago that was led by UCA&R's Dr. Karen Jetter, and you can find the details of that study on ISHB.org. So to review, as the fungus spreads, it leads to branch dieback, and it can spread very quickly in some species like box elder, which you can see here on the left. As those trees become severely infested, what we call amplifier trees, so these are trees with lots of entry-exit holes, this will be described more fully later, as well as dieback, they become huge safety hazards, as well as a source of beetles to infest other trees. We've seen some devastating consequences of this pest disease complex in Southern California. Saw it in the Tijuana River Valley, where 140,000 willow trees were severely damaged in a very short period of time. That area has since recovered, but it was a long process and required a lot of removal of other invasives such as arundo. We saw it in Laguna Niguel Regional Park, where 500 amplifier trees were removed. And we've seen it in other places, such as UC Irvine, where nearly 1,000 trees were removed, and many of the now available management protocols were developed. Visual surveys are critical to stopping the spread and managing this pest, but keep in mind this beetle is very small, and you're probably not going to find it unless you're looking very closely at severely infested trees. So what should you be looking for? Well, first of all, the term shothole borer comes from the distinctive entry-exit holes, which are perfectly round and the size of the tip of a medium ballpoint pen. Other signs and symptoms include staining, boring sawdust, gumming, and sugar-like buildups, which are sometimes referred to as sugar volcanoes. The signs and symptoms with these infestations depend on the species of the host tree, and you can learn more about those on the website. But it's very important to remember the main thing is that distinctive entry-exit hole. So if you don't find the perfectly round entry-exit hole, roughly the size of the tip of a medium ballpoint pen, it probably is not ISHB, but some other look-alike pest. On more severely infested trees, you'll also see crown dieback and branch dieback. There are lots of organizations and individuals contributing to the efforts to manage this pest disease complex, and that's a really good thing because it's a very serious threat to California's forests and woodlands. We've been very fortunate up to this point to receive support and funding through the Invasive Species Council of California and the California Invasive Species Advisory Committee. And CAL FIRE in the past has been able to provide funds for trapping and tree removals. But again, this problem is not going away, and more funds and more eyeballs are needed. So if you can help in any way, we welcome you to join the team that's fighting against this pest. If you want more information, you can visit the website. You can go to X, formerly Twitter, Facebook, Instagram, or our YouTube channel. And we will be recording this symposium as well. And you'll be able to find that recording on both the website, and we'll be sending out an email with a link to that when it's available. So that's all of my comments so far. Julie, do we have any questions at this point?
Yes, we have 7 questions. And for those of you who have questions, you can put them in the Q&A, please. The first question is there a distinction between an entry hole and an exit hole?
An entry hole and an exit hole are exactly the same thing. So the female beetle tunnels into the tree. Hole that she has dug in there is the same hole that they're going to exit out of.
Next question, do the female beetles attack only healthy trees?
No, the female beetles will attack healthy trees, sick trees. As far as I know, they don't seem to have a preference for one versus the other, but I think that some of our later speakers can probably address that more fully. We've got just a couple minutes left to answer any remaining questions.
All right, I'm going to let you do the cutoff, but I'll keep fielding questions to you in the meantime.
Okay.
Can both beetles live on the same host at the same time? And I am assuming that this refers to the two different species, the two main species, Corotio and Polygophus.
The answer is yes. They could both be on the same tree. There is no evidence to show that there's any crossbreeding between the two species. If they're both on the same tree, both of those fungal complexes will be present. So the ones that are unique to each of those beetles. One more.
Okay, have early detection, rapid response, has that been effective with ISHB?
And that is a question I'm going to leave for some of our later speakers. I think we have been successful in several cases of this, but again, it can be managed, probably not eradicated. With that, I'm going to go ahead and introduce our next speaker, Dr. Shannon Lynch. Shannon has recently served as assistant professor in forest pathology at the State University of New York College of Environmental Science and Forestry. Dr. Lynch has returned to California as assistant professor at UC Davis's Department of Plant Pathology. She also serves as chair of the California Invasive Species Advisory Committee and was last year's recipient of the Western chapter of International Society of Arboriculture's Award for Arboriculture Research. She has worked extensively in California studying the invasive shothole borers and developing a statewide strategic initiative to control and manage this emergent pest disease complex. So take it away, Shannon.
Thank you, Randall. Thank you so much for that introduction. And it's wonderful to be back in California, back in the plant pathology department at UC Davis. And I'm excited to be charging ahead full time on research for invasive shothole borer. And it's especially a critical time as we are finding new introductions in Northern California. So I'm happy to be of service here. So I'm going to be talking about some of the efforts that we have been doing to identify areas that are most vulnerable to the pest pathogen complex and ways that we can use these risk mapping efforts to basically inform efforts for control, inform strategic efforts for monitoring, and things like that. The first thing I wanted to do was really dig into how we developed the model for these risk maps, because I think that's important. So I'm going to dive right in. As Randall discussed, the complex attacks many tree species that are distributed across major kinds of systems. And those include native plant communities, as well as urban forests and avocado groves in California. And so because they are affecting all of these different kinds of systems, initially what we really wanted to do in the early stages was understand what was going on with host range, to understand spread and impact. And that's going to be really important in terms of how we decided to do this risk mapping and modeling. And previous experimental and observational work that's been pioneered by Greg Gilbert and Ingrid Parker at UC Santa Cruz, where I did my dissertation research, has shown that there is this predictable nature in pathogen host ranges, where closely related hosts are more likely to share a pathogen than those that are distantly related. And that allows us to do some really interesting predictive ecology that we wanted to actually apply to this system because of the broad host range. And like Randall discussed very early on, we observed a whole range of symptoms on a variety of tree species. And again, we also observed a lot of variation in the severity of impact, where some hosts just showed, just branch dieback, and others were killed when they're attacked. And so I'm just going to dig into this a little bit here, because in order to understand this variation across the species, we wanted to dig into the biology a little bit more and understand that when the beetle tries to attack a tree, this is really important because there's some species where it could not establish a gallery and successfully colonize on some species. While for others, the beetle couldn't establish a gallery, but the pathogen could colonize on the tree. The pathogen can't spread from tree to tree, but it could colonize. And then on other tree species, a beetle can bore into the trunk, produce a gallery, establish a fungal garden to feed on, make their brood, and then spread onto a new host. So as Randall discussed, both the beetle and the fungus must be able to reproduce in order for a host to be competent. So each of these steps have the potential to differ among susceptibility and species. And this is actually really important from an ecological standpoint and from a predictive standpoint in terms of how we assess risk. And a lot of this came from our assessment, fortuitously, some of the most heavily infested areas of the very early stages of the infestation in Southern California are home to two of the biggest botanical gardens. And because they harbor this wide range of plant species that represent unique and common ecosystems from all over the world, including those native to and planted in Southern California, we had this rare opportunity to use these gardens as a natural laboratory to investigate the plant host range of the beetle and fungus complex. And what we found was that these host types are based on these nested outcomes of interactions between the host plants and the beetles and the fungal pathogens, where the beetle makes an attempted attack on 324 species, 137 the fungus can only colonize. A subset of those 77 species are competent hosts that support reproduction of the fungus and the beetle. And those are important in the spread. And those include those 20 species that are native to California, avocado, and anywhere from 25 to 60% of trees in the urban landscape that are planted in Southern California. And then a subset of 18 species that can be killed when they are attacked. So the lethality of different hosts is important in terms of ecological impact. And I say this because this is just to reiterate a lot of what Randall covered. But what we found was that these competent host species are important epidemiologically and ecologically, because these competent hosts are important in contributing to the spread, but they also have this, what we call a phylogenetic structure. And so this is just breaking down this tree here. This thing that looks like a Death Star is simply a phylogenetic tree, or a tree of all of the relationships of all of the species, the woody species that the beetles could encounter in California. And basically what the graph depicts is this distribution of phylogenetic distances of all the species pairs that are found in California by these host categories, from the most inclusive to the least inclusive on this x-axis here. And so the distances for all of the 2,700 species in California that are represented in this, what we call this phylogeny, which is way up here at 170 million years, they're either attacked or they're ignored. But the distances for what we call these non-competent hosts, those are attacked or Fusarium colonized only species, have shorter distances than the ones that are ignored up here, which we consider to be those non-hosts. So that means that they're more closely related to one another than expected by chance. And for competent host species, there is further what we call clumping. So these are in the blue, and I kind of cartoon these in on these this phylogenetic tree here. And the competent host, there's further clumping, and the phylogenetic distances were shorter by 48 million years. And then this even narrower subset of clusters in the orange, these very closely related species, the ones that are rapidly killed by the beetle and the pathogen, they have shorter distances by an additional 45 million years. So basically what this is saying is once it's able to attack one species, chances are it can also affect close relatives with similar traits because they are more likely to behave in the same way. That's basically what this is saying. And so just to kind of summarize that, really what that graph is saying is that the hosts are not random. They're closely related. They're a subset of all of the available tree species that the beetle could encounter in California. And what that is, it's a phylogenetic signal. And that signal that there is more pronounced for each of these nested outcomes that are these interactions. And those competent host species are basically important epidemiologically, and they have a phylogenetic structure. And so what that phylogenetic part is doing is just describing the relationships. And as a first pass, it tells us what species will be affected and to what extent in novel locations. And because Fusarium dieback hosts are distributed over a complex landscape, where there are some sites that have one species, like an avocado grove, and others have a lot, like in these natural forests, what we wanted to do is use this, evolutionary understanding of host relationships to basically predict which locations the beetle and the pathogen is most likely to establish and cause damage in California. And there is good reason to do this, of course. So this is the current distribution of Fusarium dieback invasive shothole borers in California right now. So currently the red is the distribution of Polyphagous shothole borer, and the blue is showing where we have Kuroshio shothole borer. So Santa Barbara is kind of, the leading edge of the infestation. We had detected Kuroshio shothole borer in traps in San Luis Obispo, but what is most concerning just this last November was the Polyphagous shothole borer was detected and confirmed on infested sycamore trees 280 miles north of the leading edge of the infestation in San Jose. And so this is definitely setting off alarm bells, and the need to be able to have these, predictive measures is really important to be able to elicit a rapid response. So we've done the risk modeling work already, just looking at climate envelopes to identify susceptible areas. But we also know that there are other drivers, such as host availability and the kinds of species available that are important. And so what I want to explain is that this general expectation in plant disease ecology is that disease is mostly related to the density of competent hosts, which for, things like multi-host pathogens and pests, like what we're dealing with, includes other host species as part of host density. So the greater the host density, the more disease pressure that you'll see. But we also know that which competent hosts and how many of them in that kind of neighborhood influences disease pressure as well because of those host relationships. And so that disease severity is a function of host abundance and relatedness. And so what we decided to do was integrate, climate factors and host community structure to predict at a large scale which areas are most vulnerable to beetle establishment. And so to do this, we established a large plot network in infested and non-infested locations across from basically San Diego County all the way up to Ventura County. And at this time, this was spanning the entire infested range in Southern California. And we established plots in avocado groves, as well as peri-urban and wildland forests, to incorporate the whole range of conditions for which the beetle could establish. We collected microclimate data in each of these plots and measured the amount of disease if present in each. And we estimated plot susceptibility based on this interaction between, again, this phylogenetic structure and host density in a plot. So here's a plot. And what we did was we calculated the probability that each species in a local community is a host using the phylogenetic distances between it and these killed competent hosts. So those impacts were integrated into the model, and that we weighted that probability by how abundant each tree species is. And basically what we get is this logistic regression where we have the probability that a plot is infested and the observed infestation on the y-axis, and plot susceptibility based on that phylogenetic composition on the x-axis. And we have this highly significant effect in which the probability that a plot is infested increases with that increasing phylogenetically weighted host abundance. And I'll just put this here because this is another way of looking at it, with the histograms of our infested and our non-infested plots on that same x-axis. And there is a skew where the area where we predict plots to be infested are actually infested. And then more of the non-infested plots overall in reality land in those lower probabilities. So this community context alone does a really good job of being able to predict things. It doesn't capture everything because there's these sites here where we don't expect to see disease and we do. And at these sites, we expect to see disease and we don't. And so that can be an artifact of time that these sites just haven't yet been invaded. But again, we also know that climate is an important constraint on beetle development. And because we have temperature measurements in all of our plots, we could estimate the number of beetle generations per year for each plot based on the parameters from our previous experimental work and a degree day model. So we incorporated that into the model. And basically what we found was that in a plot that can produce more beetle generations, where we would expect to be less susceptible, actually becomes more susceptible. So like if it's less susceptible based just on species composition, it actually becomes more susceptible because the microclimate in that plot is more favorable for beetle development. So there's these different effects of temperature on sites depending on the community context, in which warmer places with more beetle generations of beetles allow infestations of less favorable communities. So that's how how the climate factors in. And so we took that and we want to know what is, we wanted to make the statewide predictions. So we wanted to know, is this going to take over California or not? So we applied the model that was validated from our plot monitoring data to predict infestation risk across the state using climate data and our best available data from urban forest inventories. But because the microclimate was more similar across the infested region compared to across the state, we decided to treat the effects of the community composition and host relatedness and the microclimate on beetle reproduction. We wanted to be conservative and treat those as separate but complementary factors in our statewide predictions. And so what we did was we estimated susceptibility for 170 cities in California, which have complete street tree inventories in the California Urban Forest Inventory. And this represents the largest database of individual urban trees in the United States. And those individual tree data of over 5 million trees from the inventory were aggregated into over 9,000 square kilometer grids across the extent of California. And so if we kind of zoom in, this is what it looks like. And then the likelihood of Fusarium dieback invasive shothole borer establishment was then estimated using the model and the total number of trees representing each species for each grid. And fortuitously, we have, thanks to the statewide monitoring efforts, we also have a subset of 832 grids that were not part of the model development, where other monitoring efforts were taking place that we used to test how well our model could predict establishment. And we did find that our model predicted the observed infestations of the grids pretty well. That was nice to see. And so this is what the map looks like. And what we have here is, as you can see, it's not a map, that covers the entirety of California. We don't, so this is like just a good plug for the need for street tree inventories. But what it does have is clusters of cities across the state that represent locations where the beetle could invade if the beetle was to be introduced into those locations, and locations from which the beetle could spread. And I'm just going to zoom in here just so you can see what these this looks like up close. So this is just in Orange County. And what this shows is the grids that are more yellow are less likely to be infested, let's say, and the areas in red are those hot spots. And so what our model indicated was that nearly 1/4 of these, there were 3,000 grids outside of the infested range that were actually highly susceptible. And so what that means is the beetle is able to reproduce in those areas and kill trees because the model included killed competent hosts. And so on top of this, we ran the degree day model to estimate the mean annual number of beetle generations across 20 years within each grid using DayMet data to produce a single bioclimatic map of beetle generation estimates. And this was done for grids across the state. So this filled in the whole state of California. And this is essentially what the whole map looks like. So we have our clusters of areas that are based on predictions based on species composition alone in our grids from yellow to red. And then we have the rest of the map that has estimates based on beetle generations, where you have less beetle generations from light green to more beetle generations to dark blue. And these panels on the left are just kind of zoomed in views of our maps. Areas on the left are from our infested counties, and areas from the right, I put in quotes, of our non-infested counties, because we have here San Jose, which is an infested county. And just one of the things I wanted to highlight here is that the host composition, again, is key. So if you have an area that is a hot spot, it is highly likely to be infested by the beetle, and that temperature mediates that. One thing that I want to point out is this block up here on the top right. This is Sacramento. I just wanted to point out here that the climate is much more suitable for a beetle infestation, and there's a lot of hot spots in here. And so this indicates, this is indicative of areas that we probably want to really focus monitoring efforts just to keep an eye on for an infestation. So basically, the map looks like this is what the map looks like when you combine community composition and the bioclimatic map, and it illustrates how it can be used to determine which locations are more susceptible than others when you use this kind of host composition and the climate in combination with one another. And essentially, the model and the map can be used to identify locations where control treatments will be most effective and help reduce the use of pesticides, and also inform where to strategically remove trees and reduce costs. And one of the things that I just want to also point out that we are doing to kind of improve the performance of this map, the important thing I want to highlight here is that the map is a really good first pass at being able to determine, where the beetle is most likely to go, given whatever information that we have at this time. And it is going to continuously be refined over time. And one of the things that we're working on is integrating landscape factors, as well as the way that spatial dispersal factors as well. And that's going to be important in improving the map as well. But so far, it does a pretty good job at predicting things, and it's a good first pass. And so I just want to thank everybody, all of our funders and all of our collaborators that have contributed to this work and putting this huge effort together. And I look forward to taking any of your questions.
Dr. Lynch, how can land managers use your density phylogenetic model to develop a monitoring program for early detection?
Yeah, so basically, we are in the process of making the map available. And we're working on that so that it can be used as a tool, as an interactive tool to basically inform your monitoring efforts.
Related question, is the model searchable for cities which were included or have inventories in the California Urban Forest Inventory?
So I think to basically protect privacy, I don't think that is an option at this stage. But once the map is available, I think it's easy to kind of hone in on specific locations to be able to kind of determine where you want to focus your efforts and things like that. One of the things that I want to invite people, because we're always we want to improve this map, is if you have street tree inventories, we would love those inventories, and to incorporate those and build those into the map as well. So this is definitely a plug for more inventories so that we can actually expand the power of this map as well.
Did your models include competent host density and the phylogenetic relationship of the hosts as separate metrics, or did you use a phylogenetic diversity metric like Faith's phylogenetic diversity?
The former.
All right, I think that brings us exactly to where we're supposed to be at this point. Our next speaker will be Dr. Beatriz, or usually referred to as Bea, Nobua-Behrmann. She is a University of California Cooperative Extension Advisor for Orange and Los Angeles counties, specializing in urban forestry. Her research and extension program is focused on finding appropriate management strategies for urban forest pests that affect Southern California trees, like the invasive shothole borers and the gold-spotted oak borer.
Thank you, Randall. Hopefully you can see the right slide.
All good.
All right. Well, thanks for that introduction. And we're going to dive into what are our best management practices that we can use to take care of invasive shothole borers and Fusarium dieback infestations. So a quick overview of what I'm going to be covering. I'll be talking a little bit about integrated pest management and how to prevent pest problems. Then I'm going to talk about monitoring and how to do survey and trapping for invasive shothole borers, management plans and management matrices on how to, what to consider to make a management decision. And then I'm going to dive into the removal of amplifier trees, how to dispose infested material, and chemical management options. So let's get to it. Everything I'm going to be talking about is within the context of integrated pest management, right? So this is a kind of strategy that focuses on long-term prevention of damage, right? So it's not something like, it's not a patch. You want to make sure that this is something sustainable. And so for that, we use different kind of practices combining cultural, mechanical, biological, and chemical. But as you can see, the way we use them, there is a preference, right? So we try to use cultural management first, then mechanical, then biological, and chemical management is kind of our last resource. The idea is to use pesticides only after we know that it's like we monitor and whatever we find indicates that we need it, and we do it based on some established guidelines, right? And we only target our organism or pests of interest. The idea with this is to minimize any kind of risk to human health, to non-target organisms, and of course to the environment. So because of that, prevention is basically one of our main tools in here. So just to go over some of the basic prevention tactics, we're talking about good cultural practices, right? So we're talking about good irrigation. Generally, in all these landscape trees, the prevalent problem is either too much or too little irrigation. And especially when we talk about trees that are in this open areas like parks, where it's like turf around the trees are getting frequent and shallow irrigation, right? Which is exactly the opposite that most trees need, which is infrequent and deep irrigation. And also, the other thing that is important is to consider the timing of the irrigation, right? Because if we're here in Southern California, our native oaks, for example, they're adapted to the summer drought and they do not need to be irrigated in the summer. So, and if we irrigate the oak in the summer, we're creating all this moist in the base of the tree that then makes it prone to some fungal diseases and root rot and other stuff. So proper irrigation is one of the main ways you can enhance your cultural practices and prevent pests and diseases in your trees. Of course, another one is soil preparation. You know, if we have soil compaction is really bad for our trees. We can add soil amendments only when it's necessary. And something important here, people want to make their trees more healthy and say, oh, let's put a lot of fertilizer in it. Really, mature trees rarely need any fertilization. And actually, when we over-fertilize, we can make those trees more susceptible to pests. So beware of those packages of, oh, here's your treatment for your trees, and I'm also adding all this nitrogen fertilizer to make it stronger, because usually it's not needed and it might be detrimental. Then tree selection. of course, is another cultural practice that we can use to prevent pest problems. So always finding the right tree for the right place, so we don't have to over prune it. For example, if there is a power line, we don't want to plant a tree that grows too high, because then we're going to have to top it or like prune it very heavily every year so it doesn't reach the power lines. And then, yes, plant selection, especially when we are replanting, it's important. Try to pick some of the species that are less susceptible, but mostly when we are planning on planting or replanting, the most important thing is to aim for increased diversity. We want to avoid any kind of monoculture, because that is like opening a buffet for shothole or any other pest. So here is a whole 3 blocks of the same species where you can come and infest them all, right? So really trying to not have monocultures, having things more diverse. And when you're trying to think about replanting, the first thing you have to do is check on what you have, right? What is already in that area, and then pick something that complements that, right? And ideally something that is not so closely related, because as Shannon said, we have for shothole at least, the preferred trees are all, you know, closely related to each other. So just try to make your palette of trees, especially in urban trees where you have a lot more possibilities to pick from, just try to make it as diverse as possible. And then lastly, you know, preventing the spread of the pest is always the first step. So that means dispose correctly of any kind of infested wood, and I'm going to be talking about that a little bit more in depth later. And disinfecting your pruning tools after you use them. This is not necessarily for shotholders, but yes, for any kind of fungal or bacterial pathogen. So when we're pruning a tree because they have a branch that has dieback or you know, like, we need to just cut that branch, and then we go and trim another branch with the same tool. We are being the ones that inoculate that other tree with the fungal pathogens. So we are being basically kind of like the beetles, right? So always, if you can, disinfect your tools between prunings, but at least disinfect your tools between sites. So at least you're not bringing the pathogen that is in one side into the other side. Ways you can do this is using alcohol diluted at 70%. That's because diluting it at 70% makes it a little bit runnier, and it's easier to you know, clean the blade. You can also use it at 90%, but at 70% is what you can find anywhere, and it works really, really well. You can also use a solution of 5% bleach, which works as a disinfectant, but I would not recommend that for any kind of blade, because bleach will rust your blades really quickly. And in a pinch, you can use Lysol or some of the other disinfectants to spray on your tools. But I will always recommend that if you work as an arborist or a you know, tree pruning company, always have some sort of way of disinfect your tools in your truck. So just a spray bottle with 70% alcohol, it's ideal. You just have it in there. Whenever you need it, you just spray your blade and let it air dry. So now we're preventing pest problems, and then also we are monitoring, right? We want to know when we find these beetles in this beetles pest complex is present, right? So Randall did say, like, you need to know when it's there, you know, to find it pretty early. So monitoring for it and finding it early and figuring out what's going on as early as possible is one of the keys to successfully manage this pest. And for monitoring for shothole borers, we have both vision inspections and trapping. So when we do visual inspections, we are going to look at the trees. We're going to look for these signs and symptoms that Randall already covered. We're going to be especially looking for this entry hole, perfectly round and about the size of the tip of a medium ballpoint pen. And to do that, we're going to be looking at all of the trees or all of the host trees. Ideally, you will still look at all of the trees because you could find trees that we don't know their host yet. When you observe your trees and you're looking at the trunks and the branches of your tree, usually there's more infestations on the north and east facing sides of the trunk. It's not always the case, but is the most common case. And so make sure that you go all around the tree and you look at the tree from all angles. When we do surveys, we use binoculars to be able to look at the top branches and see if we can see those holes, or in a lot of the cases, especially when we're looking at it high up in the canopy, seeing the holes is very hard, as you can imagine, but we rely on those other companion symptoms, especially the staining and the staining or the gumming. So make sure if you see some staining on the top canopy, then use your binoculars to try to see if they seem to be associated with some holes, and you might either need to get some kind of platform to go up and look at those branches. And then once you looked at the tree very carefully, you can assign an infestation level to that tree. And for that, we use both the number of active entry-exit holes and, if we observe or not, dieback associated with the infestation of the beetle. So we will say that a tree is likely infested when it has less than 50 exit holes. We'll say it's moderately infested if it has between 50 and 150. And this is within one main trunk or one main branch. A tree is heavily infested if it has more than 150 active entry holes. And we'll consider it severely infested once it has more than 150 exit holes, entry exit holes, and we observe dieback associated with this infestation. But I don't know if you noticed, I was talking about active entry exit holes. So I want to talk a little bit about that. What is an active entry hole versus an inactive entry hole? This is something that we've been sort of realizing as we were surveying more and more. And this is something that you most probably find in areas that have been infested for a while. And that's why we find this here in Southern California, for example, in Orange County, where I do most of the tree monitoring programs. And what happens is that a tree could have an infestation, and later on, either the tree responded to it and was able to compartmentalize those galleries inside the tree, and so basically the fungus is not expanding and the beetle is dead, or the beetle died because of some treatment or whatever other reason, and so that hole that connects to a gallery is no longer active in the sense that it doesn't have a live beetle inside. And so the things that we notice to distinguish an active versus an inactive hole is that an active hole will be the very well defined, the roundness of the hole, which in inactive holes it starts to getting sort of deformed. You still see it weeping, or you still see frass coming out of it. That is a very good indicator that there's a live beetle inside. And of course, many times you can see the beetle abdomen sticking out of the hole, which is, of course, a sign that there's a beetle in there. For the ones that are inactive, you usually see it sort of a dry hole, like you see here on the right side. In sycamores, you can still see the hole, but but it's surrounded by this like gray tissue. There's no weeping, there's no frass coming out of it. And sometimes, especially in London plains, there's like this bulging that happens where the hole used to be, and the hole is somewhat deformed. That's basically what happens after the tree compartmentalized around that wound. And if you have any doubts, which we had a lot at the beginning, you can always do the paint test, right? So for the paint test, what we do is we cover the entry hole with a layer of water-based paint, just the same one that you will use for your walls. And then usually you come back like a couple of hours later, or you can come back the next day, but within what is a matter of a few minutes to a couple of hours, if the beetles are inside that gallery, they will reopen the entry hole. That beetle does not like to have that entry hole closed, and so she will reopen it. So you'll see the hole again open. That is an indication that there is a live beetle inside that hole. But if this hole is still closed, it tells you that there's no beetle in there. And we've done this with a lot of holes in a lot of trees, and I can tell you that when the hole looked like this on the right side, it never got reopened. And when the other thing to consider when we are surveying is which species of trees we're going to be looking at, right? We have our 65 species of trees in California that get attacked and can be reproductive hosts of invasive shothole and Fusarium dieback. That is the same as Shannon called them competent hosts, right? So we have that list. We can find that list in ISHB.org. But also, even within that list, in a lot of the cases, the beetles are not able to colonize that tree unless it has a previous fungal infection. So keep an eye on those trees and keep an eye on other species that are not in the host list, but you still see that the beetles attack. So I'm gonna go back to what Shannon was showing. Just remember that she showed that in some cases the beetles might attempt an attack and fail, right? Well, if you are in an area that is highly infested, it's very possible that you'll see that in a lot of different hosts, in a lot of different species of trees, right? When there's a lot of beetles in an area, they start getting adventurous, if you want, and they start trying to attack all different kinds of species of trees. And I would not just dismiss it because that tree is not within the host list. I will keep an eye on it. Look for those holes. Do they look active or not? Is that infestation or the attacks in that tree increasing? And it shows like there's more and more and more. Because what happens sometimes is that the beetles keep attacking a tree and they're not successful, and that tree does not work as a reproductive host or a competent host until, for other reasons, that tree gets attacked by, let's say, bud canker, right? And in this is the case that happened with the Casuarinas in here. So this is a Casuarina. It got some sort of wound, and it got here a Botrytis infection, and we started finding beetles. The beetles were attacking these Casuarinas, but not being able to establish. But in this particular tree, they started being able to establish here at the margins of the fungal infestation, and after that we started seeing some trees have a few hits, but nothing, none of them seem to be successful. But eventually we found trees that were sort of like in the category of moderate. So they have like more than 50, like 100 entry holes, and that just rings some alarm bells. And we went and checked, and they were felling one of those trees, and we were able to get into that wood. And sure enough, we found galleries and larvae of the beetle. So be aware of those species that, or those trees that might have some fungal infection, because they could become a host for the beetles in those situations. The other thing you can do for monitoring is trapping. So the idea is to use trapping as a complementary method to visual inspections. We can use either Lindgren funnel traps, or the ones that are more preferred lately are the sticky panel traps. And there's different kinds, so we can use here at the very right the big elm bark beetle traps, which are pretty big. And it has been tested, this one's against smaller size of this sticky panel trap. And so you can use a bottom century wing trap, or you can use a medium size, which would be 2 of these bottom century wing traps back to back. And what we found is that these 2, the big one and the medium ones, they don't differ in how good they are for finding or catching beetles, but they are better than the small one. So either of these 2, small or medium, tend to work best. In all the cases, either you use a small, a big, a medium, or a funnel trap, you have to use a lure. So for lures, we use cruciferol. We use the one that lasts 1 to 2 months, and we only use 1 lure per trap. That is important because if you put more lures in a trap, it will deter the beetles instead of attract the beetles to the trap. And something I want to bring up here is that this lure is not a pheromone, right? Cruciferol is basically is a smell that the trees make. And so it's kind of indicating the beetles, oh, here's a host that I might like. So it's not really super attractant as a pheromone will be. And for that reason, you can put the traps fairly close to each other, like 10 to 15 yards apart. But also, it won't really attract beetles that are very far away from that trap. So you will be trapping whatever is within that location. That means that using traps will not bring beetles to your property if the beetles are not already there, but also means that you cannot use the traps as a control methods because they're not attractive enough. So what do we use the traps for? We use them for early detection. We use them for guiding our inspection efforts. So in areas where you have the capability of using a lot of traps and you want to be managing, it really helps to have traps around because they will help you focus your visual monitoring efforts. In some of the parks that we work with, we have the beetles sort of colonizing the top branches of oak trees, and it's really hard to see them from the ground. But when we put traps, then we can know that there's a certain area where the traps are catching a lot of beetles, and we put more efforts into looking into those trees, or even sometimes, getting some kind of cherry picking or platform so we can go look up into the canopy of those trees. Another use, you can put them in the borders of your property and see if the beetles are coming in from outside, and you will know where are they coming from. And the other thing you can use them, and it's very useful for that, is for helping you time the treatments when the beetles are dispersing, especially if you plan on using some kind of contact spray insecticide. So you want to apply it when the beetles are actively flying and actively landing on the trees. And here is typical patterns that we find here in Southern California about trapping. So this is the percent of beetles trapped caught in the traps in the different months in 2 different parks. But as you can see, even though we do have 2 peaks, one in the spring and one in the fall, in both cases, in the top one in Whiting Ranch, we have the spring peak happens a little bit later than in Irvine. So there is variation among sites, and there's also variation among years. So if you have a trapping program in the place where you're managing the beetles, you can find this curve, and as you can see that the beetles are starting to fly more and you start to catch more beetles, you know that it's time to, if you are going to do some kind of treatments, you know when exactly will be the best time to do treatments in your location during that year. So for deciding what management strategy to use, we developed this management matrix. You can find this same table here within our pest note that is available at ishb.org. So for this management matrix, the factors that we consider are the value of the tree. If the tree is low value, we go to this top one, and if the tree is high value, we go to this lower one. So what's a high value or low value tree? That kind of depends on the property manager, but generally high value trees are bigger. Low value trees might be in poor health, and usually the other thing that we tend to consider high value trees are those heritage tree, those historically important trees as well. But it will really depend on who's asking, right? Because if you have 3 trees in your backyard, they will all be high value trees. So does the high value or low value will depend on the land manager's decision. And then within that, we have the host type. So if it's a reproductive host or a non-reproductive host, the hazard level of that tree, right? If there's branches that could fall on people or property, and then the infestation level that we already talked, that you will be assessing during your monitoring. And so as you can see, depending on all of that, these are different broad recommendations. So in general, for low value trees, we tend to recommend monitoring, and then once the infestation gets into moderate or higher, we recommend either removing those infested branches and sometimes removing the tree. And remember, here we're talking about low value trees that might be small, unhealthy trees, but because of other reasons, right? You can have a tree with a hole in it, and it might not make much sense to go all at it when the tree is structurally not very good and it won't last too long. And then for those high value trees, you can consider some treatments for when the tree is moderately to heavily infested. Sometimes you can consider treatments in low infestations, but generally, if you manage the moderately and heavily infested trees, you can probably avoid treating the lightly infested trees and see how that progresses. If you keep monitoring, those trees that are lightly infested, once they don't have too much beetle pressure, they might recover on their own. Lastly, what you can see is that if the tree is a non-reproductive host in either kind of value, we only recommend to monitor. You would not be treating those trees by any means. And if you find those trees to be moderately or heavily infested, then we ask you to notify us or the ag commissioners, because that is a tree that we might go look at, because it could be a new host that we didn't know of before. Also available at ISHB.org is this sort of decision-making tree that is covering similar information as the matrix, where you can make a decision based on where the infestation is located. So if the infestation is in the branches, pruning might be a better option. If the infestation is in the trunk, and if it's highly infested, you might need to remove that tree, right? And that's what it says in here is once the infestation is severe, removing the tree and the stump is what is recommended in all cases. And that is because when you have a severely infested tree with all this dieback, the chances that this tree is going to recover are very low. And if it has a lot of the trees, it says more than 150, but we can have thousands of entry holes in these trees. And that means that you have thousands of beetles potentially leaving that tree and infesting all the trees around it. So in this case, these are not the most common, especially if it's an infestation that is getting managed at most you find one or two. When you're not managing an infestation, then you have more and more trees that end up in this severely infested category. So managing early is very important. And so these trees that are far too gone and they're probably not going to recover, we consider them severely infested and we recommend removal. Also, these trees can have so many galleries in their branches that they can become very brittle and they become very hazardous. And when we do remove those trees, we're left with a stump. And that stump, and I'm not sure if you can see here, in this case, there's still a bunch of entry holes in this stump. And beetles can still live in these stumps for a long, long, long time. So the recommendation is to also either use a stump grinder to remove the stump, or if you cannot do that, I will flush cut the stump, as low as you can, and then if possible, even cover it with dirt. The other thing we can do is remove heavily infested branches. So that is especially a good idea in some of the species of tree like oaks or avocados, where the beetles tend to attack most of the branches and not so much on the main trunk. So in that case, you can just remove the branches that are severely infested, and you don't have to remove the whole tree. You can keep the tree and just remove the infested branches. When you do this, it's really good to spray the pruning cuts with Bacillus subtilis as a biological fungicide to prevent fungal infestations in those pruning cuts. And remember that dressing pruning wounds is something that we shouldn't do anymore. And these heavily infested branches, is important to remove them because when you have that, you can also have some secondary pests that come along. For example, and this is an oak, a coast-live oak that had this very heavily infested branch, and it was flagged for removal. A few months passed and it didn't happen, it wasn't removed. And then we went back and we started finding Western oak bark beetle and foamy canker in those same branches that were debilitated by the shothole borer. So these holes here are shothole borer, and this one that looks like someone spit on it is the Western oak bark beetle with the foamy canker. So we are enabling some secondary pests to also grow and possibly infest other trees. So once we remove the trees or the branches, it's very important to manage that infested plant material. The ways to make sure you're not going to spread this pest through the movement of infested firewood or infested green waste is to chip that wood as ideally on site. Chipping as small as possible If you can chip to less than 1 inch, that is ideal. That will get rid of 99% of the beetles. But if you cannot, because I know it's really hard to get that small of a chip, chipping as small as possible is always a good idea. Chipping your material already reduces the beetle load in that material by a lot. And you can use those chips as mulch as long as you keep them in the same property. Please do not move those chips to a different property or a different area to be used as mulch. If you want to use those chips in a different area, you have to either solarize them or you have to compost them in order to make sure they're pest-free. And lastly, if you want to use the logs, solarizing the logs or kiln drying the logs will get rid of the beetles. But you have to do that before you even think about moving those logs elsewhere or using them as firewood. Now, if we want to consider chemical management, you have to keep in mind that we're only gonna be considering treating the trees that are highly infested and high value. There's no preventative treatments that we know work. So doing preventative treatments is just a waste of money, and it's putting chemicals in the environment that don't need to be there. In general, we save those treatments for moderately to heavily infested trees, as I said. The lightly infested trees, you could maybe hold on treating and just keep monitoring and see what happens. And when we do treatments, we usually use a combination of insecticides and fungicides to attack both the beetle and the fungus at the same time. Make sure you consider, where the tree is, how healthy, is the tree worth it to be treated, or should we just remove it? What's the level of infestation? What is around that tree? And we're gonna go into that for a little bit more in a second. Make sure you have a licensed person applying the pesticides, because everything that we tested that worked is only available to state licensed and county registered pesticide applicators. And always remember, no treatment works 100% of the time. So just treating your trees is not gonna get rid of the problem right away. So you have to keep monitoring and make sure that whatever you did worked, and if not, you have to adapt your management and find another way. Options, we have chemical management options are one of them is trunk sprays. So we can spray the trunks and the bigger branches with bifenthrin, which is an insecticide, and Bacillus subtilis, which is a biological fungicide. The idea is you will be killing the beetles that are leaving the tree to disperse, or arriving into the tree to attack that particular tree, right? Like they came from a different tree and they're landing in your tree. So this is a contact insecticide. So you should apply it when they're dispersing, and you should be careful because it's not a targeted insecticide. So basically any other insects that would land on that tree will also probably be affected by these pesticides. So one of the things that is important is to avoid spraying to flowering plants or flowering trees, because we don't want to affect especially because we don't want to affect our bees. And something very important too is to not spray when you are close to water, because this pesticide bifenthrin is very detrimental to aquatic organisms. So we don't want any of this drift to go into any kind of body of water. The other option is to use systemic pesticides. So these are taking up through the trees and they kill the larvae inside of the tree. They do depend on tree uptake. So basically we want the trees to be well irrigated. Ideally, we'll irrigate them before and after the treatments. And the best time to apply them is during spring or fall, when the trees are actively taking water and it's not too cold or too hot. Options for that, we have soil injections or soil drenches with imidacloprid. Considerations for soil injections are, you just have to make sure that the soil is not too compacted or it won't work. And again, because it could leach into the water, you will avoid areas close to the water. And our other options for systemic pesticides is trunk injections. So for that, the pesticides that have been tested and worked are emamectin benzoate and propiconazole. So a combination of both. So you have an insecticide and a fungicide again. And for trunk injections, you have to consider that it requires drilling into the trunk. So you're making a wound into the trunk. So you have to really think if it's worth it or not, because that wound can get infested by fungal pathogen, and then you have even worse problem in your tree. But the good thing is that lasts 2 to 3 years, so at least you won't have to do it so often. But be very careful, because secondary infestations of those injection sites, as you can see in these pictures, is pretty common, especially here in Southern California, where there's a lot of Boschosphaeria spores floating in the air and easily can colonize those wounds. One thing you can do to sort of help prevent that kind of fungal infestation in the wounds is to spray those injection sites. After you inject, you will spray those injection sites again with Bacillus subtilis as a way to help prevent infestation of those wounds. And something that is just in the development is using spot injections. So basically that means using a syringe with an epidermic needle and injecting, this is 0.1 to 0.2 milliliters. So like basically literally a droplet or two of the pesticide directly into the entry hole of the beetles. So we tested this and it works. We tested it with emamectin benzoate, imidacloprid, and acephate, and they all work better than the untreated control. So it is a good option for a tree that will justify a very labor-intensive management option, right? Because you have to go and inject every hole. But if you have an area, if you have like a few trees and they have a few holes and you want to make sure they don't get infested, that's an option. Or if you have a high-value tree that you want to protect, that's also a good option for that. And as I said, just look at where your tree is and what you can do if you are going to be treating. Because if you're in this place next to water, your only option will be to inject that tree. Or if you have a tree like this one, a soil injection in this medium here with all this compacted soil and very little soil available will also be very hard. But because it's in a parking lot, maybe a trunk spray might be your best choice in here. Even then, what I want to pass on is don't apply pesticides just because you have an infested tree or because you think you have an infested tree, right? So make sure you go through all the tools in your toolbox first. You might be very well by just removing a branch, or just wait and monitor that tree and see if the infestation progresses or if it doesn't. So don't consider chemical management as your first choice. Will be there. So in conclusion, Shothole is here to stay, at least in Southern California, but there's no need to panic. Make sure that you take timely action, and Shothole can be manageable. And so make sure you educate yourself about Shothole and other pests and diseases and how to prevent them, and take those preventative actions so you don't have problems with pests and diseases. But once you have them, look for science-based management options and use all the other tools in your toolbox first, and leave chemical management as a last resource for managing this and other pests. And with that, there's a lot of people and organizations that help get all the information that I just presented. And here's my contact information in case you want to contact me. And I don't know if I have any time for questions.
So here's the situation. So our schedule has you ending right now, and we have a break until 10. So it's up to you. Do you want to take questions while we have the break? People can go on break, or do you want to answer questions later?
I don't want to deprive people from their break. I'm okay if people want to ask questions, or I can take one or two now and then answer everything else in the chat if that's preferable.
Great, Dr. Bea. We have quite a few questions, so I think your suggestion to answer a couple of now and maybe as many online as possible might work. If imidacloprid is used, do we need to also use a fungicide?
Ideally, that would be good. So a lot of the times we combine the soil injection with imidacloprid with a trunk spray with bifenthrin.
Do you know if any research has been done with regard to treating the pest disease complex with bark banding?
Yes.
Not that I know. I mean, let's see if Dr. John Kawashima here knows about it. Bark banding.
So that would be bark banding with dinotefuran. That has been tried by applicators, and unfortunately, we don't have any scientific data. When Dr. Tim Payne was initially doing the studies, he found that dinotefuran was inconsistent. Sometimes he'd get good results, sometimes he wouldn't. The people in the field are saying that if you do it repeatedly, which is very expensive, but if that's something that fits into your situation, it's possibly effective. But from what we've heard from arborists is it takes repeated applications for it to really be effective. Some of us have thought that a soil injection or drench with imidacloprid along with a bark banding is something we should look at in the future when we test. And the reason for that is that imidacloprid moves faster in the tree than dinotefuran in terms of solubility. But dinotefuran imidacloprid soil drench might be like a 1-2, where the dinotefuran, because it's a bark banding, might be quicker. And then the follow-up would be imidacloprid being picked up by the roots and going into the tree. So it's a interesting option to think about. We haven't really done a test of that, but it's something that's worthwhile looking at in future trials.
Is there a minimum age or tree part diameter that shothole borer will attack?
That's a great question. So we have seen them attacking basically all sizes of trees. However, they are more prevalent in trees that are at least 15 inches in DBH and higher. But I have seen them in younger trees as well. It's just less common. They tend to prefer trees that are medium to bigger, but it's not impossible. We've seen very small trees also get attacked.
I've also heard with respect to that the tree host makes a big difference. That, for example, if it's box elder, they don't really care what the size of the DBH is.
Well, absolutely. There's a big difference between how likely a tree is to get infested based on the species of the tree, right? If it's a box elder, and so if you have a box elder and there's beetles in the area, it's only a matter of time that they will find it, and they actually will attack it and probably kill it very, very quickly. Then, at least here in California, the next ones in the list, at least in my experience, tend to be like the sycamores. They get attacked pretty quickly. Like it tends to be also one of those trees that you can see that like as sentinel tree, if you want, not as much as a box elder, but a tree that when you're looking at an area that you didn't know of any infestation, you will definitely have to look at your sycamores, because they might get infested pretty quickly. But for a sycamore to die from shothole borer, it might take 3 to 4 years before it dies, or even longer. Willows also get attacked pretty quickly in riparian areas. Yeah, willows, sycamores, and box elders, I'll say, are within the 3 species that get attacked quicker and more intensely.
Thank you. Yeah, let's go ahead and if people need a break and
I'm
want
sure
to get
people
straight
need
to the
to.
questions, let's everybody take a break. But let's get back at 10 for our next speaker.
Sounds good. I'll answer any questions you guys have in the Q&A. So if you have any follow-up questions, you can leave them there or send me an email. Thank you.
Okay, well, let's go ahead and get started. Our next presenter is Kelly Parkins. She's the Plant Health Care Coordinator at West Coast Arborists. Her work in the turf and ornamental markets for the last 40 years has included arboriculture, golf course and sports turf construction and management, technical sales, agronomic consulting, IPM program development and management of grounds maintenance and park development in the municipal government sector. I guess there's not much that she hasn't done. She's conducted more than 300 workshops to industry groups and professionals on various aspects of horticultural systems and management.
That introduction, I had forgotten that I'd done some of those things. It's good to hear that. All right, so actually, I'd like to say I think it's a great speaker panel today. I'm learning a lot. I agree with what Randall said at the beginning. Just when you think you know everything, or maybe have the problem licked, we get our hats handed to us. So I'm going to go through some of these challenges that we continue to have. From the perspective of a pest control business, West Coast Arborists is obviously a tree trimming and planting and tree management company, and we're one division within that organization. I'm the coordinator, so a lot of the work I do is assessing pest issues for our mostly municipal, school district, county clientele, and then developing programs to address the problems. So I'll be going over the evaluation of the challenge. Mostly these are urban forest hosts across the state of California and into Arizona, implementation of treatment programs, and then assessing those treatment programs by way of monitoring. We use monitoring as our primary method of working through these challenges, the pest challenges. We also use tracking systems with inventory software systems and inventory specialists to do that work. I'll be going through that. And then looking at the effectiveness of treatment protocols, as well as some of the pest regulations, pesticide regulations that we're facing now. So I've been involved since 2021, but West Coast Arborists has been managing these systems in Southern California since 2017 as kind of a turnkey operation, assessment and treatment. In Northern California, infestations were found earlier this year. We have not done treatments up there yet, but have clients that have host trees, so we'll be working with them as we go through. I will say that the focus of my presentation today is really looking at some of the surprises this year on ISB infestations, both the treated and non-treated stands, and the number of tree species that I'm seeing these on. And a note of some of the perspective on the agencies that we deal with. You know, many of these are public agencies with tight budgets. Their treatment programs are sharing budgets with grid trimming and inventory and planting. So assessing these pest programs is one facet of it, but really trying to get the budget to work for some of these treatments is a challenge as well. And then Bayer kind of indicated this as well, as pesticide use is not hopefully the primary resource. I'm going to talk about treatment programs of what we use, but there's a whole a bunch of tools that you can use, and we need to use those because residents, the public is around those areas where these pesticides are being used. So it's really a toolbox approach, improving the pest control of the susceptible species. So the IPM management tools that Shannon has talked about, BEA, most of these have been developed by UCCE, the Cooperative Extension. We use their matrix as our field guideline. So when I'm out in the field, I'm assessing trees with that kind of in the back of my mind as I go through them, trying to assign trees to monitoring programs, monitoring treatment programs, follow-up programs, that type of thing. So we use that toolbox exclusively. The foundational component of our program is survey and monitoring. We go out in the field first. We inventory the trees. On the monitoring, we're looking for those compartmentalized wounds or the galleries or active infestations. Dry hits, as Bay indicated, are great indications of the tree managing this pest, or that the programs we put together and utilize are also managing the pest. Wet hits are good indicators of an active infestation. And then I use the canopy as a critical component when I'm evaluating the trees. If I have a tree that has a fairly high or say moderate level of infestation, the canopy is in good shape. It's a full canopy with little impact from Fusarium. We look at that factor as a determining factor for treatments. So level of treatment we choose can be based on the health of the canopy. And treating with moderate levels, you know, this is kind of my assessment from my experience in the field. I feel like low and moderate levels of infestations with full canopies are good candidates for treatment. And a key point that Randall made is that the insects don't feed on the wood. They're farmers. They're cultivating the fungus that's living inside. They're living in the tree, cultivating that fungus. So we're trying to affect their ability to cultivate the fungus as our end goal. So an overview of the Southern California region. I again will state that I think the treatment protocols are very effective in managing the population. There's been an arc of treatment protocols over the years. They've done a good job at investigating treatments and then refining them to produce a better outcome. Urban forests are typically what we're managing. Parks, I'd say, are the lion's share of the type of area that we're managing. But we do also wildlands, state forests, open areas in terms of managing these pests as well. And those challenges can be very unique. It is hard to get into the area to monitor the trees. It's unlikely you're going to be frequently back to that area, and that can predispose you to, that's going to color your decision making in the treatment that you're going to choose. And I'll give you examples of that as I go on. My assessment is if you're monitoring and treating and following your IPM toolbox, most of the trees on these programs do not progress to a higher level of infestation. And then most populations we see tend to be low levels because we're in these areas performing different types of work frequently. So we're able to catch it, I think, when the populations are low. So typically pyrethrins with the biofungicide. We use a penetrant and then an extender now as well, kind of added that back into the mix, which I'll go over in just a minute. But I think that's working well to keep the populations low. And I'd like to also state that 100% control is not, I don't see that as a possibility, but keeping the trees healthy and the aesthetic in our areas high, I think is the goal that is very achievable. So by way of kind of showcasing the work that we do and kind of the approach we take. I have two projects that we've worked on to highlight. I have a new monitoring and assessment that we did in June of this year, and an ongoing assessment that we've done since 2017. So we're in our 7th year with that project. Okay, so this is like I said, a new project that was presented to us. This site was inventoried, and I went out to the site after it was inventoried. There's 271 trees, and many of these trees are ISHB hosts. 30%, after my evaluation, 30% of the trees have mostly low-level infestation, but there are some moderate and some heavy. But the fact that was really interesting to me was how many different species were affected. So cottonwood, sycamore, alder, box elder, California buckeye, Sonoran palo verde, and coast live oak. So a lot of different types of trees. I'd never seen it before on the palo verde or the buckeye, but it was really interesting. The other thing that was really interesting is most of the trees had stunning canopies, so really full canopies, even though they were impacted by the ISHB. So I'm going to go through a couple of the pictures I took. This is the California buckeye. You notice the canopy is impacted on this tree. This one is scheduled to get a trunk injection because of the level of infestation. It's a heavy level infestation, and canopy is beginning to pull back. And as we go through July, August, and September, as the months get hotter, this is likely to become more pronounced. Coast live oaks, very low infestation, full canopy. This tree's in very good health, so we expect a good result. This is box elder, which is like ice cream to the ISHB. There's a lot of box elders on this property, but if you look at the right-hand side view, the canopy's in very good condition, so we'd have an expectation of a good outcome. I don't find many white alders anymore. I think the Phytophthora hit those 10 or 15 years ago. You don't find them very much anymore, but these definitely have ISHB, but again, full canopies. So as with the California sycamore, we expect a pretty good outcome. I've never seen it before, like I mentioned, on the Sonoran palo verde. This tree has somewhat of a thin canopy, but I know the rains and the cooler temperatures may be a factor with that as well. So we'll be watching this as we go through. And then kind of as an example to show you how I use our system, any tree inventory system that allows you to take photos and incorporate that into the information on the site information is really valuable. So I went through and took pictures of all the trees that were infested. Those are uploaded to the site along with any of the work that's been done on the site, whether it's tree trimming or the treatments that we're preparing to do in this next week. All of that can be loaded onto the site. And this is valuable. If you're inspecting thousands of trees a year, it's hard to exactly remember what they looked like during the initial inspection. So this gives me a chance to document that in the inventory system. So even though these trees appear to be highly infested, most of them are low level. They're just very, very a lot of weeping. And again, I don't know. I'm not sure if that's related to the amount of rain we've had in the last 2 years. I'm not really sure, but most of these are below 50 hits. They're low level. Like I said, about 30% of the trees are infected. So we are going to go ahead and treat these trees, mostly with bark sprays, to inhibit the spread of the insect and fungus that the insect pervades. There are 8 trees that are heavily infested. 50% of them, which is not unusual, are box elder trees. So we plan to do trunk injections on these trees that will quicken, hasten the time period, the period of time to get the insecticide and fungicide into the tree to begin to control both the insect and the disease. But this next project, the second project, has been an ongoing project since 2017, as I mentioned. We've done a number of monitoring projects in these locations over this period of time. The most recent was done in fall of 2022. We looked at 863 park and street trees across this city. And I'm going to talk about just this specific, a single park site overview as it relates to the work that we've been doing. So there's 246 sycamores, mostly the race Mosa, which is the native species, and 125 coast live oaks. Now, Bea talked about the monoculture issues with this pest, and it is an issue, I will say. Not that we don't want to plant sycamore trees, we do, but an overabundance of one species really makes this more difficult to manage. So after doing the evaluation of the trees in the fall of 22, we treated them the following year in 2023. About 34% of those were treated. That's probably an average over the last several years. And you'll see what I mean in just a minute. But so we have had success since 2017. We've lost few trees. The treatments have worked well. So I went out in the winter of 2024, kind of expecting the same thing, and this is what I got. So this was kind of a shock to me. And the tree on the left, I walked up this park from the south, so I was feeling pretty good about myself until I got to the north side of all these trees. And the tree on the left and the tree on the right are park trees there. The tree in the middle, I put that picture in there to indicate really what the dry hits look like. So you have this compartmentalization of the wounding by this insect. And that is mostly what I have seen across this project for the last few years. This year was really an exception. And I will tell you, I really don't know what's going on. I haven't seen this level of infestation in the 2 monitoring cycles I've done in this city. So not sure what's going on, but the tree on the left is this is our inventory system. We can see here that we've been treating this tree since 2017, 2018, 2020, 21, 23. So regularly treating the tree with a number of different types of treatment. But we've monitored twice. I have a mistake there. This tree wasn't last tree in 21. This is the last tree in 2023. The dry hits are obvious on the lower trunk part of this tree, but we are currently scheduled to do a trunk injection just because of the severity of the infestation in such a short period of time. I was out on this site in the fall of 2023, following up on our work that we did in the spring of 2023 on the treatments, and did not see any of the wet hits that are present now. So it was a very short period of time that we've had this kind of turnaround. This is another tree at the same location. This tree is kind of hard to see, but a lot of sycamore borer, kind of that roughening of the bark on the lower areas. But if you get up in these areas, you can see the infestation hits. Maybe it's kind of hard, but I really wanted to leave this as a wider view to see that there is canopy. And you may say, well, Kelly, that looks pretty thin, but the canopy, it's hard to tell from this picture, has really been impacted by anthracnose. We've had almost 50 inches of rain in a very short period of time over a 2-year span, and anthracnose is a really big problem on a few of the landscape trees here in Southern California. So this tree again has a severe infestation level. I'm sure if I were talking to Bayer or John or Shannon, they would maybe indicate that the tree should be removed, but the fact that it still has produced a significant canopy, we're going to trunk inject the tree with emamectin benzoate and propiconazole, and then I'll be back there frequently to monitor the effect of those treatments. Again, not trying to beat a dead horse, but it really, you can really see that more of the older hits on these trees, a lot of compartmentalization, which is what I've found repeatedly. It gets to the upper portion of this tree, and looking back into the pictures from previous evaluations, this section of the tree had not been infested with this pest. So this is a kind of an outbreak in the upper part of the canopy. It was a moderate level infestation in 2021. Fall of 2022, like I said, you really didn't see an infestation in the upper canopy. It wasn't treated in 2023 because all of the wounding was old. They were old hits, kind of compartmentalized. And come back in 2022 for the evaluation, nothing there. 2023, no treatment. And in 2024, this winter, that's what I came upon. So we have scheduled this for a trunk injection as well. I think this is kind of a downer, showing all the things that haven't gone as we had hoped. But this tree, you can see extensively treated for a period of 7 years. The last one was in 2023. In 2024, I came back and the tree was dead. This is certainly a severe level of infestation. This tree was removed last month. So which is really the best thing, is remove the stumpless ground so that the infestation can't spread to the other trees surrounding it. Okay, so I'm going to move through some of these next slides pretty quick. Bea went through the matrix. That is the matrix that we use as well. The parameters that most guide us, there are factors in the toolbox that we pay attention to, host, infestation level, value, and hazard. But primarily, we are not doing risk assessments out there. We're doing general healthcare assessments. What we are paying attention to, is it a reproductive host or not, and what's the infestation level when we set up the training. The inventory system, you can see how valuable that is to be able to track what is being done to the tree. The other thing is you can also put in different asset classes into our system if you want to specifically track a certain pest. And again, these treatment protocols were reviewed already. I'd just like to say that in 2023, the pest note on ISHB went from we used to treat no infested trees, the no infested trees we treat, and that moved to monitor only. But like I said, there are times that we're treating, for instance, if we have a park where we have a large number of reproductive hosts with high value that have no beetles and no population, but it's surrounded by trees with, say, moderate infestation. And we know that budget constraints are going to not allow us to get back out to do treatments for a period of time. We may elect to do treatments or recommend to our client that they do a treatment on trees that are not infested as a protectant. So this is kind of a game time decision, boots on the ground. We don't make these decisions. We present the information to our client, who's the owner of the trees, and they will make the ultimate decision. So I'm going to scoot past this as well. This is the tree treatments that we do. So trunk spray, soil drench, soil injection, trunk injection. And I agree, trunk injection can leave the tree open to pathogens. Bacteria is one of them. Before we drill into the tree, we sanitize the drill with alcohol or Lysol. do the injection site. Post-injection, we treat the open wound with the Bacillus subtilis product. So that has seemed to work very well. Part of the challenge of making sure the treatments are working is getting back out there. So I typically go back to each site to look after about a 6-month period to document the status. And frequently I don't have the time to post those pictures of the trees there, but the next monitoring cycle that we go out on, we will. And like I said, reproductive hosts, if there's non-infested trees in the area that weren't treated, we do definitely want to go back and look at those trees. And then I will state that the program that UCCE has put together, really, if you're monitoring and treating, the success of this program is really awesome. I was a park superintendent prior to this, and I took out 1,000 sycamore trees that succumbed to this pest, kind of prior to the protocol being available. And I think it's great that we actually have a way to provide care for the trees and keep them. Kind of in closing, I do want to say a couple of things about the pollinator protocol. The insecticides are pollinator disruptors. You cannot use these around foraging bees. That's one consideration, because we're the pest control business. We have to make sure that we're adhering to those rules. In many cases, the trunk injections are done because of that. The ivermectin benzoate is not a pollinator inhibitor or effective pollinators, so that's a good choice. And then the sensitive environments, most of our clients are public agency, and people using these sites can be particular about the use of pesticides. So we're cognizant of that and try and make sure that the programs that we're working on with our clients work to the advantage of how the public views their management. The other thing is, I'll say a little bit to the groundwater and surface water regulation. The BEA indicated it is toxic to aquatic organisms, so using it near bodies of water is problematic. But in most cases, you can safely apply to trunk sprays to most trees without any issues. And that was pretty quick, but that's it. I'll take questions, or I can look through the Q&A also when I'm done and answer any questions that are in there.
Yeah, we've been trying to answer questions in the Q&A online as we've been going along. We're actually right on time for our next speaker, so let's take that approach with the few remaining questions that are in there. And our next speaker is Rhonda Wood. Rhonda is a seasoned urban forester based out of Orange County, California. She dedicated 25 years of service to the Disneyland Resort, caring for over 16,000 trees. She is ISA certified as an arborist, as well as a certified tree worker. Rhonda is passionate and committed to serving as a leader and educator in the tree care industry. She spent countless hours volunteering as an instructor for the WCISA's Women's Climbing Workshop and served as the WCISA board president. Oh, she's also taught arboriculture courses at the college and university level, is a tree risk assessment qualified through ISA, and a graduate of the Municipal Forestry Institute, as well as Consulting Arborist Academy. And she and her husband are now embarking on a new venture, launching a traveling consulting arborist business. So, Rhonda, please take us on the next part of this journey.
Thank you, Randall. So good morning, everybody. I wanted to provide a bit of what we've learned when we worked at Disneyland for over 25 years. And we at first, of course, as everybody, when they first discover that they may have an infestation of invasive shothole borer, we were, of course, very disturbed by this and scared for what was to come for Disneyland especially. But luckily we learned a lot of lessons along the way, and I think the resort is better off because of the beetle, because it actually taught us how to inspect for pests and disease and manage it in a sustainable way. And so that's what I'm going to walk you through today. So we're going to talk about our best management practices, which is much of what Bea spoke about and what Kelly is using is the approach that we developed as time went on. And I'm going to tell you how we did that, and then just provide some concluding thoughts if we have time. So 2015 is when we first discovered ISHB in infestation. And so we found out we had an early infestation from Dr. Keith Escalon and Dr. John Kabashima. So we did a lot of investigating, scouting for this beetle and figuring out what we had. And of course, if anybody knows me, I'm always smiling. And in this picture on the right, I am not smiling because, as I said, this is something that scared us, and that we could lose. How many trees could we lose at Disneyland, and what would that impact be? So when we realized we had this infestation, we have 16,000 trees, over 680 species of trees. At first, we thought we were immune before we found out, because experts said, well, you have a diverse canopy. Maybe that's why we're not seeing any beetle activity, because we did monitor before 2015. We found one hit in 2012, couldn't find anything else. So we thought, again, we were untouchable somehow. So when we learned we did have it, we did everything we could to educate ourselves. What are they doing about the beetle treatment? And so we went to UC Irvine, where the UC Cooperative Extension, UC Riverside was conducting research on how to treat these trees. And in one case, they were doing trunk injections, which was, again, just a trial at the time. And so we were just learning as they were learning, but we needed to react, right, to prevent a full-scale attack. Through that, we learned from the experts how to develop a coordinated approach for monitoring ISHB. We needed to request funding, because this is something we didn't have. We weren't looking for this beetle, and now we needed to figure out how we were going to fund inspecting for this beetle. And we, of course, consulted experts, and we sought all sorts of in-depth training. We went to every seminar. We went to as many, like, can we just tag along with what you were doing, like we did at UC Irvine? And it also helped to befriend the experts. Now, of course, Disneyland has the benefit of great attractions that we could lure them in and have a little fun along the way. But anybody can do this, because I'll tell you that these two in particular can be lured by food. So Dr. John Kabashima, if you give him food, he will come to your place. That is the key to success. You don't need an attraction. And so through that, again, we're educating ourselves. We came up with this best management practice, which is first early detection, and then we'll talk about trapping and scouting. In learning what we needed to do, we did the preliminary traps with that were a little bit more complicated, and I'll show you that we went to sticky traps, which were a lot easier, and were like the key to our success. And the key to Disney's success right now is the traps. But lessons learned. So when we first discovered it, again, you think Disneyland, well, all trees are pretty visible, but we found our infestation occurring on the berms, which actually have limited access. They're surrounded, they're what screens Disneyland around the entire property. And So it's very dense, and it was hard to see this kind of infestation. So this is an amplifier tree, a coral tree, and that was our biggest infestation was erythrinas, had the most activity and were one of the amplifier trees that we had to remove. So the amplifier, again, we had amplifier trees, we didn't even realize it, and this was spreading across the resort. We didn't have traps at that time, and so the first method of defense was what do we have? What is our situation? So the traps were telling us where the beetle activity was on the property. And so we would scout areas with high trap numbers. We also had a database that of course told us what tree species we had and where they were. And so we used that to also monitor those trees for what kind of beetle activities they had. And that took us quite a bit of time to go through the entire 16,000 trees with the species we had. And we were still learning at that time. They were still learning from a research perspective what species were being hit. So, like I said, it's a trap. The traps were what really told us where activity levels were. So we put over 100, at least over 100 traps, I think, surrounding the resort, so we can see where the beetle activity was coming into the resort. And then we also put them on the inside of the resort. And we found out the jungle cruise, which this trap here shows on the left, 600 beetle hits in the jungle cruise. So we knew we had a heavy infestation in that very moist, humid area. And that's where the beetle comes from, and that's what they like. So sure enough, yes, they were in the jungle cruise. So then we developed a monitoring program. And at one point we were monitoring all tree species that were susceptible every month. So we can determine what level of infestation we had, how many hits they were, are they low, medium, high, and make decisions on what we do in terms of treatment. So this is back in 2019, like we were monitoring 2,200 trees. And I think at one point we were monitoring about 3,000 trees before the activity level went down, which I'll share with how that happened. So like I said, the monitoring became our method to control the beetle activity, to manage it. And so we located where all the trees were. We acquired the tools necessary to inspect for the beetle. We trained our team members to monitor and look for the beetle, decipher if it was something else other than ISHB. We started looking. We then tracked this in our database. And so these are the tools. The training, we trained not only the tree assessment team that we had that typically did risk and health assessments, but we also trained all the arborist team that was climbing and inspecting these trees on a regular basis, pruning, because in a lot of cases, they're the ones who discovered up in the canopy where the beetle activity was happening. Of course, our IPM team was trained, and we even did outreach with our gardeners, of course, management, the management team, consultants and researchers were all learning at this time. And so what we were looking for, I'll show in a minute, like we looked for everything from, at first it was like, well, we were looking at the lower level trunk, but we did canopy inspections. We found as little as, 1-inch branches that were reproducing with the beetle. So it was a very wide spectrum of what we were looking for. And in one case, we were looking at stumps because there was a lot of activity on stumps that we didn't even realize we needed to look at. And as Bea showed, we would use the paint method, and this video shows how you can see the beetle butt moving. Once we painted that, immediately comes out and wants to open up that hole. And so we used, of course, our climbing team, our arborists, we taught Akif how to climb trees. He knew how before he had ropes, but we taught him how to climb safely, I would say. And so we went up in canopies and looked for this activity. In one case, we utilized drones, but drones are very difficult because of the, you need the qualifications for drone activity, but we did have, we sought resources to potentially budget for drone activity. Disneyland is not really the best place because we only have a short period of time of daylight before the park opens, and in some cases we barely have any. So it didn't become a method that we used very often. And we would sample activity and send it to the lab to make sure what do we have. We found we both had Polyphagous and Curasiro. And then at first, before we always had a database to track our trees, but what we didn't have was something that was designed for inspecting invasive shothole borer on the monthly basis that we were, and providing information and utilizing it as a tool to help manage decision-making on how do we treat this pest. And so we had a spreadsheet, which was crazy. People were printing out spreadsheets and writing on it and walking all over the resort, which wasn't effective. So we had to adjust our database to match the need. And so we did that, and that was great. So that is one thing to consider, is your tools being a database. And if you don't have it, of course, a spreadsheet is possible. It just makes it a little bit more challenging, and you need more resources to manage it. So the map-based approach was really easy for our teams, including our arborist tree workers out there, so that they can see which trees we were talking about that have the beetle activity and communicate with each other, as well as having a system that, you can document, like Kelly said, having pictures and being able to communicate with the tree assessor to the arborist climbing, which branch am I talking about? And we were able to include pictures in the database, and they could just look it up right there as they were going out in the morning and pruning the trees. And so with one click of the button, we could see which trees we still needed to monitor that month, of which trees had susceptibility to ISHB. And so our team members would just go to each of those dots and inspect them, say what level of activity there were, and then we would follow up with what would be the treatment level for those trees. And then the database also had a capability of historical reporting, and that way we can look over time and see how we were trending, which was really important to also be able to budget for what we were seeing, and then how we were able to control it as well, providing that data. The IPM team would actually go take traps every month and count how many beetles there were, and then we'd be able to see whether it was high, medium, low activity, and then scout in those areas. So mitigation, mostly we found that we could prune out branches that had invasive shothole borer. That was our primary hope versus using a lot of treatments. But of course we did use treatments. Also, so the target treatment was based off of the species, the environment. We would use injections, of course, near waterways, difficult situations like that. But one thing I didn't mention on our lessons learned in the very beginning was that when we first launched on this activity, when we didn't have the help from the research team completely with the program, we injected over 600 trees immediately when we found out we had invasive shothole borer. Just mostly at the time, Quercus and Platanus is what we knew we had a lot of. And so we injected those. And what we found was that by injecting, we caused cankers in some cases on the trees because of maybe poor methods of using the injection systems. There are better now. But we caused cankers, which actually cause issues, obviously, with other diseases on trees. And so we went to this monitoring method, which helped prevent just preventative injections that actually caused more problems, and they were good. And of course, we did have to remove some trees. I'd say overall it was in Disney's case, because it's a show-based area, that we removed trees that maybe didn't need removed, but because of their aesthetics, we removed them. And so it was probably only a couple hundred trees that we had to remove. We focused, of course, on the amplifiers, and I'll talk about replacements as well, but stumps became something we had to document, which we didn't prior. And so we actually had a documentation on all our stumps, and we started removing stumps because of that project. Disposals, following all the disposal, of course, we didn't want to spread more shothole borer across Southern California. So of course we would chip appropriately. And then our reforestation plan was, as we always had, we want a diverse forest. So we continued to diversify. We had a naughty list of trees. So basically, because we work with landscape architects, Walt Disney Imagineering, we needed to educate them on what trees we shouldn't plant at the time because of this beetle unknown, how we can control it. We were still in that stage of how can we. So eventually that naughty list, it would adjust. It wouldn't just be completely never plant a platanus again. We were able to, once we were able to have some management, some control over the beetle, we would be able to allow some planting of platanus in certain areas, but we would only want what we could manage. And so we would work with Walt Disney Imagineering to do that. And we would also, we were working on an urban forest master plan at the time. And so that information was always, we wanted to document this process. So we'd always be able to do this management plan. So from a reforestation example, we actually utilized our back of house area where we have 30,000 employees, and about at least half of them would cycle through this back area where there was a big administration building, and we had Coleritarias that were reproducing. Invasive shothole borer was reproducing in these trees despite their sapping, and they would enter through canker. They were canker associated reproducers. So we had 53 Coleritarias in this area, way too many of one species in one place. That was mostly the species that was in this location. And so we removed those trees because treatment wasn't effective, and we replaced with a diversified palette that was non-susceptible to ISHB. We did Torrey pines, ginkgos, and a few other species. So we went from one species to like 6 species in this area, which was better for this area. So here's just an example of what was, and the dieback that you see on those Coleritarias, and then what we planted. And yeah, so that is bringing it all together. This was the method to our madness, which took time to get there, and we learned a lot along the way, and I believe it or not, we had fun doing it. I can't believe I could say that, but it's true when you develop a system and you keep up with that system. So now they use the traps to monitor the trees. They don't inspect every species monthly. They use the traps to scope those areas out if we have any higher activity. And their method is prune. They do, of course, some treatment, pest applications, but we try to minimize that, remove amplified trees, which they haven't had since 2018 or so, proper disposal methods, and then a reforestation plan. So just some concluding thoughts. Of course, you might all think Disney has the budget. Of course, you can control this, but that is not always the case. That it is a corporation, and we do have, shareholders to please, and so it was a big undertaking to ask for funding to pay for this. And so Dr. John Kapashima actually helped us with a lot of the data on what would happen if all the trees that were susceptible in Disneyland, if we lost them and had to replace them, what would that cost? And of course, for anybody in finance to see that kind of number and realize, wow, if we were to actually replace all those trees at in a particular size, and they would never match the same size that we would have to remove, but at least I think we estimated 48-inch box trees on average. That what would that cost? And it was a ginormous cost, and so that's how we were able to get our funding. And so that every organization has a few of these folks, and so that's who you need to convince. You can, this is a spreadsheet that we used to be able to fill in that information and share what the cost would be on a program. And it was obviously a lot less than replacing thousands of trees. And so that was a big success. We were able to increase the amount of people monitoring our IPM program headcount, and dedicate an arborist team, an inspecting canopy arborist team to inspect for beetles and treat for them. I think from that standpoint, these are the big takeaways. Know what you're looking for, know what you have, organize your team, find the support, the budget. You have to scale and prioritize to what you have. And you can do that in the interim, even before budgets are increased, if that's possible, and then make it sustainable. So, and again, I think the best thing that we did was collaborate, and you don't need a Disneyland to collaborate with the experts out there. The UC system has been, the UC scientific team, as we called them, were outstanding in their level of help and assistance. They want to see the best for California and for actually internationally, as I know that many of them have traveled and helped share how to control this beetle. So with that, I just want to thank Dr. Keith Escalon and Dr. John Kapashima, and many others that helped us along the way, helped us learn, and for not just invasive shothole borer, but for the many other different pests and diseases that we have out there, a method to help manage it. So that is all.
Thank you, Rhonda. So we don't have any specific questions for you, but a great presentation. I think you need to be moving on to something else. So thank you for being here today, and we'll move on to our next group of presenters.
Thank you, Randall.
So next up, we have a roundtable discussion from the leading edge, if you will. This is a group of speakers who have been dealing with this issue in some of the areas where we're right on the edge of where this pest is going and has been. Stephanie Stark, Deputy Agricultural Commissioner with Santa Barbara County. Of course, Santa Barbara County was where the leading edge was prior to the more recent find in the city of San Jose. Jocelyn Perez, forest ecologist with the Inland Empire Resource Conservation District. And Jocelyn, I don't know if you saw, but we did have a question in the Q&A related to Chino Hills State Park. I don't know if you'll be able to either comment on that here in this discussion or directly in the Q&A. Sarah Davis, City Forester, City of San Jose. Drew Raymond, Deputy Agricultural Commissioner with Santa Clara County. And lastly, Igor Lacan, the Interim County Director, Bay Area Environmental Horticulture and Urban Forestry Advisor, Cooperative Extension for San Mateo and San Francisco counties. So he'll be wrapping things up, talking about some of the outreach in Northern California beyond Santa Clara County. So with that, we'll go ahead and start with Stephanie.
Thank you, Randall. This is Stephanie Stark with the Santa Barbara County Agricultural Commissioner's Office. Thank you very much for allowing me to join you today. This is a really exciting topic, and Santa Barbara County for quite a while was the leading edge for the state. So I am sorry to say we have lost that title and the pest has moved on up north to San Jose area. I'll just make this really brief, but we kind of thought that it would hit our county really hard. It had hit Ventura County. We're adjacent to Ventura County, but it didn't hit us as hard as we had expected. So maybe that could change, of course. But I was just going to show you a little bit about our approach and what we took. I won't take but more than 5 minutes. I was just going to talk a little bit about our partnerships, what worked, what were some challenges, and kind of what we're doing now. So our program consisted of, it was mostly the grant from CAL FIRE and CDFA, and we were tasked with really doing mostly outreach and removing the worst of the worst, so the amplifier trees. We didn't do treatments or anything like that, but the grant was really focused on the work in the field, trapping, as well as doing surveys and then any removal. And we followed all of the UC guidelines and what Bea had talked about earlier. In all, the grant was 4 years or almost 5 years. It was 5 years, and we removed 47 trees. Most of those were sycamores. We did also have some London planes and some mimosa trees, surprisingly. Overall, there were 31 trap locations countywide. We had 75 traps placed. We kept them all year, around all year. And we had found Caricio as well as a polyphagous shothole borer. The first site we had was in the Montecito area, and that's where the Caricio was. All the other locations we found to be polyphagous shothole borer. We focused mostly on public safety and trees that were in that area. So the tree removal, we didn't do tree removal in backyards like some counties had, but we did focus on tree removal on public lands or quasi-public lands. For example, parks, private and public parks, tribal lands, golf courses, cemeteries, open spaces, institutions and nonprofits like the Botanic Garden, Lotus Land, UCSB, the University of California, Santa Barbara. We worked a lot with landscape professionals and arborists and collaborated with over 30 different agencies and departments. So we did, I think, 2 different symposiums for them. And we also worked with both the California Department of Food and Agriculture Lab, which we have an established relationship for all of our pest prevention programs. And they were able to do a pest ID on whether or not it was ISHB or not. But we also worked with Dr. Stouthammer's lab to get it down to the species to determine if it was either a polyphagous or crucio, because we did have both. We wanted to see what the differences were where they were showing up. So we would do wood samples as well. So what worked is it appeared that removing some of those amplifier trees, especially in the Montecito area, and that's in the southern part of the county. And partway through our program, we started finding it in the San Ynez area, where actually there's a fire there right now. But it is the middle part of the county where all the vineyards are. We had found it in a private park, or sorry, a county park. And we found that removing the trees, when the amplifier trees, when we continued, because we'd continue monitoring throughout the years, and when we put out traps in those following years, we did see a reduction in the ISHB populations on those traps. Successful relationship building helped a lot, especially getting out those partners to the field and seeing what the disease trees look like. And also those little UC kits that we got that had the little wood sample with the pen, and I think it had a field guide that had all the different trees and the hosts and the symptoms that you would find on these different trees, was very helpful. We also did a lot of tailgate trainings with partners, and early engagement with our county planning department helped to pave the way for the removal of the trees. We also worked a lot with California Department of Fish and Wildlife. So we had limits on what we could remove and when. And so mostly our tree removal season was from about September to maybe January, when there was no bird nesting season and we didn't have to get special stream bed permits. We worked a lot internally. We kept spreadsheets, had annual lessons learned meeting, and also obtained, what people have talked about today, the importance of tree inventory lists. And we did get those. That was very helpful. The one issue we did have with that was the proprietary software that a lot of different agencies use. So it made it hard to share some of that data. But we didn't have an internal GIS system, so we used iNaturalist to do our tracking of the trees and the photos and the mapping. So some limitations, as I had talked about, was removing the trees in riparian areas. One thing was hard to do was the stumps, because sometimes the stumps could not be removed and taken out of the creek because that would destabilize the creek banks. So that was one issue we had. We'd cover it with dirt, but of course, a creek, water will come through that. But we did inspect those stumps to see if they did have ISHB holes. And that was something that we did also. Determining whose property it is was always fun. And sometimes we just didn't remove a tree because we ran into those issues. Some of the city jurisdictions just didn't understand the timing and the pressure we were under to do removals. So we ran into some of those problems and tried to work them out. One of the challenges was green waste. So understanding the stream of green waste in our county was a challenge. It's complicated. And we had tried to find a local compost facility that would take the chips. We always double chipped all the infested wood, but getting them to a location at our county wasn't possible. We had to take it to Ventura County, but it was an approved facility. Some of the surprises were that the insect had jumped to San Ynez, and we never found it in Carpinteria, was directly adjacent to Ojai, which was very infested in Ventura County. And then I mentioned we had both species. The drought made it really difficult to see some of the staining and gumming and on the symptoms. And also there were impacted mimosa trees. So that's it. And I didn't want to take all that time. So if there's any questions, but I'm on the panel, so I'm happy to answer any questions.
So let's keep going with our next presenters. We'll go to Jocelyn next, and we'll allow time at the end for you to answer questions or have a discussion among you. So Jocelyn, you want to tell us about your experience?
Yeah. So I'm with the Inland Empire Resource Conservation District, and similar to Stephanie, we were also grant funded through that CAL FIRE grant for invasive shothole borer surveying. And we really just wanted to know the presence of invasive shothole borer within San Bernardino. So we did both trapping and also individual tree surveying. And we wanted to be like strategic about where we were placing traps. There's difficulties in doing individual surveying, especially in places that are so dense in like riparian areas. So we placed traps in places that were dense along riparian areas, and then we also did individual surveying in green spaces, parks, golf courses, etc. So I think in total it was around like 100 trap areas that we placed, and then surveyed over 8,000 trees. Over 200 trees we found with infestations of invasive shothole borer with varying degrees of infestation. I think working within a conservation district, because we're non-regulatory, we can't enforce any kind of management, and I think that's one of the big challenges that we had. We're also restricted to working within our boundaries as a conservation district, and of course we know there is no boundaries when it comes to invasive shothole borer. So that was definitely something that's, hard to work with. And yeah, and I would say another difficulty is that whenever we would find infestations, depending on the city or the municipality, there'd be like varying degrees of how receptive these land managers were. We did have successes with like working with City of Highland to get some trees removed in their park and open space. So I think definitely the collaboration was a success in working with partners and doing outreach events. We would speak to universities to just raise awareness, and I think definitely like showing those samples and talking to the public. And going in the future, although we're no longer grant funded, IRC will continue to do surveying, and especially through our trapping network. I think just recently like found out about an infestation banning, and that kind of triggered us to be like, okay, so what are we going to do? And we talked to wetland conservancies, and they were concerned with their trees in Oakland. So in the coming weeks, we were able to figure out, okay, what are the next steps? And now we're going to set up some traps there too. And then same thing, I think there's concerns with state parks. And last year we set up traps, and because we are within, we have to work within our district boundaries. That means that only part of the park can be monitored by us, and then another part of the park can be monitored by state parks. And we were able to train staff to do their own monitoring, and we provided, all the information. We use field maps to do these surveys, and now they're gonna try to expand their trapping network to possibly other state parks and other areas in Chino. So I think that's been probably the biggest success, is like working with partners.
All right, thank you. And now on to the current ground zero, Sara Davis.
Yeah, hi. Thanks for having me. I've been in this role for about a year and a half, and we are building a program from the ground up in terms of our urban forests. I have experience with being ground zero for emerald ash borer in Colorado, so I understand how this should work and how it should go. However, we still have our regular work to do. So we have rolled out kind of a bare bones trapping plan. So we're just trying to figure out the extent of what our infestation is, so we can figure out exactly how big it is and how concerned we should be. We did find the borer outside of one of our riparian corridors in a street tree, which resulted in a removal and a replanting, which cost the property owner about $5,000. So we're looking at this more as a public health social concern, more so than an insect or a fungal issue. So we're thinking in terms of multiple layers. So our stormwater permit is coming under scrutiny in terms of federal and state fines because of pollutant loadings into our riparian corridors because of our unhoused population, which happen to be around all of the trees that are the amplifier trees. So our box elders primarily in our riparian corridors. So we're having to think about doing removals around people who were basically evicting out of the places that they've been living in. So that's a challenge. We have a hiring process issue. We have identified an individual that we wanted to bring on as an intern who has a recent graduate degree in entomology, but because of our hiring practices, we can't bring them on as an intern. So we're having to explore different ways to bring this individual on to be our trapping coordinator so we can figure out, again, the full extent of our infestation. There's just a lot of moving parts involved in being the municipality that's dealing with this issue. If it was just a straight up insect problem, great. That would be easy to handle. But there are so many people and so many layers of jurisdictions involved that we can't really do anything until we have that figured out. So we can't do removals of amplifier trees because we don't know who the trees belong to. In some cases, they belong to our water authority. In some cases, they are under the ownership of private property. Sometimes they're under the jurisdiction of our parks department. Sometimes they fall under the Department of Transportation. So yeah, it's great. We can talk about the pathology of the whole problem, and it seems really cut and dry, and we can talk about treatments and non-treatments and how to approach these things, but our real focus is dealing on the human aspect of this problem.
Thank you for that. That's really interesting. So on to Drew Raymond to talk about what's happening beyond the borders of San Jose.
Yeah, hi Randall. Thank you for having me in the discussion here today. Thank you, Stephanie, for your presentation. That was very helpful from another agricultural commissioner's office's kind of point of view there. We haven't done any real boots on the ground work. We've been doing some surveying. Kind of like what Sara was saying, we don't know how far spread the beetle is, so we're trying to do some delimitation as best as we can. It is in some parks. The beetle is in some street trees, but it seems to be going through the couple riparian corridors that we have that go through San Jose. Like she said, the biggest hurdle to doing survey work is the homeless population, unhoused population in our county. It's where the beetle is, there's a lot of unhoused people in the creeks and in those areas. So it's a little dicey to do some survey work. I think that for whatever reason around maybe our office, we work in like a light industrial area, and there are some green waste companies that move green waste around. So I think we are kind of ground zero right on Oakland Road, if you know San Jose at all. As far as our ag department's concerned, or as far as what we've been doing a lot of outreach, focusing on outreach. I did a presentation to the Vector Control District a couple weeks ago. They're in and out of riparian areas all day long looking for mosquitoes. And so for them to just kind of look for some of those obvious signs of staining and gumming on trees, I thought that would be pretty advantageous of us to do that outreach. We're also trying to do public awareness. We are going to work on a press release to kind of make the public aware. I don't know if the news will go with it or not, but we'll see. Just to kind of spread the word, because we don't know what we don't know. So we don't know how far it's spread throughout the county. I believe Sara has traps. And then I believe from our last stakeholder meeting in the northern area that Mid-Peninsula was going to put out some traps too, and they're more of a woodland area, so it's kind of a different ecosystem up there than like street trees and urban landscape. But we'll see what those results have. But we've been telling people in our outreach, like if you have anything suspect, bring it into our office. We'll submit it to our CDFA laboratory. We'll get a positive identification on it. Again, we're still kind of in that new stage. It is kind of nice that other municipalities have done a lot of work and a lot of research on control methods that we can share with other municipalities and landscapers and arborists and other cities and stuff like that. So we're primarily focused on outreach right now and communication and education.
Fantastic. You want to wrap this up, Igor? Tell us about the outreach that you've been doing.
Yeah, just very briefly, a couple of things. I've been speaking to water districts, park districts, cities about this. I've been speaking to cities about this for about 10 years and was getting kind of a tumbleweed rolling past the audience response because we didn't have it. So now there's a lot more interest. So some things that have worked very well, for example, are those kits that you sent us, Randall, and specifically that little piece of wood with the holes, because then we can find the nearest wood pile, and this is something we just discovered. Is there a wood pile somewhere? We can go to the wood pile with the little piece of wood and compare and contrast all the insects that live in wood, many insects, and you can actually see the difference in hole size between our usual suspects for boring beetles and the shothole borers. So even just having that reference piece of wood has been very, very, very helpful. And getting everybody oriented to, okay, new pest, new problem, here's what you should look for, exactly as Drew described it. That's been very helpful. And then getting, and again, something Drew described, getting people who don't manage trees, so your public works crews that come into contact with trees, vector control agencies, again, people who come into contact with trees but don't manage trees, getting them oriented to, look, this is a new problem, at least for the foreseeable future. If you see this, let us know. Ultimately, once it's established everywhere, we're not putting additional work permanently on your plate here, which is important. We're simply asking you to tell us right now. We're in this kind of awkward stage. So that's been one thing that's worked well. The other thing that's worked well was to sit down with, especially agencies that have a dog outside. Maybe you can hear him.
Is that a detection dog?
Not my dog. What's worked well was to sit down, especially with agencies that own land or cities that manage a geographic area, and then do a kind of conceptual analysis. One is, we know how this thing is going to be spread. A lot of it's going to be, for example, in dumped wood. Do you experience any wood dumping? And then people, oh, yes, we do. Okay, where? So that's your conceptual pathway. Then geographically, okay, where does the wood get dumped? Okay, here's some of our roads where wood gets dumped. Here's some of our, ranching access where somebody who is camping might move firewood, things of that sort. The firewood movement seems to not be on people's radar as a major point of infestation. So that's been something we've kind of discovered around here. Less maybe of concern to a city, more of a concern to a land management agency. And it's been interesting to see the interest go from 0 to 100 in like 6 months. I was hoping this wouldn't happen during my career, but here we are. So, happy to entertain any questions. Happy to participate in discussion.
First of all, of the panel itself, do you have any points you want to talk to each other about, questions about others' experiences, or where we should go from here?
Well, I think my initial frustration was around disorganization, because who's in charge of this thing, as there's not really one regulatory authority that's supposed to really take charge of this urban incident. So I think we kind of freaked out a little bit and called in CAL FIRE, and they've been putting together an incident action plan. So we've been trying to draw together all of the cast of characters that need to be involved and treating this the way that firefighting agencies do, as kind of as an incident command type structure. So finding the right people to sit in the right chairs in terms of surveying and detection, outreach and education, and logistics and planning, of all those having some sort of a liaison person to interact between all of the different agencies involved. So that's something that we've been working on, which may or may not be unique to this sort of pest response.
Anybody else?
Yeah, this is Stephanie, Santa Barbara County Ag. I just wanted to also say to the folks in San Jose, if I attended a couple of your initial meetings, and I'm happy to attend more of them, and or talk, one-on-one about any ideas or bounce some, thoughts off me, or I can do the same. So I'm happy to be supportive in that regard. And then I forgot to mention about what we're doing now, because our grant ended. And so the pest is still here. And what we have done is we have transferred a lot of our supplies to, like, for example, some cities, and UCSB will be taking on some of our traps. I gave them everything that we have, because we no longer have funds to, continue the level of trapping we were doing and pest identification. Although we will continue to do outreach with the public, and we will continue to identify and send pests to the California Department of Food and Agriculture lab and determine if it is ISHB or not. And we'll support in that realm. As far as what to do after that, we're not continuing to monitor, but I'll still gather and reach out to our partners and let them know, where we are in the process through emails and what have you. Maybe we could do like a group call or something with our partners, is what my thought was. But it's kind of tricky when your grant ends. And perhaps we could, seek more funding. I know that that's always an option, like with the SISAC is doing the rapid response grants that they have available for counties and for pests such as this. But just wanted to mention that.
I think along the way, there was a discussion about, well, why do we care? What do we need all this information for? Why do we need inventories? Why do we need test and monitor? It's because if we don't have the data, we can't make the budget requests to deal with the problem. So in terms of being practical and being, a municipal forester, I need to know how bad something is or could be for the next budget cycle. And I need to be able to start building the momentum behind the issue to see if the citizens actually care enough to put money behind the issue. So, and it's just interesting that all of this coincides at the same time. All of the Inflation Reduction Act money is hitting. So we all have these massive projects going on with a different focus, but then we have this huge problem potentially that's about to, break crests on the beach. So there's a lot of things swirling around all at once, and the resource unit is always the dollar.
Well, we're about out of time. Julie, do we have one or so questions?
This one's for Sara. What public messaging are you doing with regard to ISHB in the San Jose area? And what are you asking of community members?
Nothing right now. We haven't been able to get in front of the mayor to let their office know exactly what's happening just yet. That meeting is coming up in August. So once we're able to brief the mayor and the city administrator of what we would like to do and get their feedback and direction on that. We're just kind of in this holding pattern. And again, we don't want to cry wolf. The sky is falling when we don't really have our hands around how big the issue is just yet. So we're just kind of in this pre-planning type of a phase. I'm not comfortable with it, but that's just the reality of where we are.
We have a comment from one of our attendees who says he doesn't feel like there's adequate time to ask you all the questions that he has, which is very true, because we're going to need to move on to our final group of speakers. I would say that if people have additional questions, please do put those in the Q&A, and hopefully the panelists will be on for a little bit longer and may be able to address some of those questions through that mechanism. But I'll go ahead and introduce our last group of 3 speakers. Shannon's already been introduced, and so I don't think I need to introduce her again. We also have Dr. Keith Eskelin, plant pathologist, cooperative extension specialist, and lecturer in the Department of Plant Pathology at the University of California, Davis. You've seen him mentioned in multiple presentations already. His primary research interests include detection, biology, epidemiology, and control of fungal pathogens on grapevine, fruit trees, small fruits. He earned the award of Arboriculture Research by ISA in 2016 and an Outstanding Education Material Award with his book chapter in California Master Gardener Handbook in 2015. And he also served as president of the American Phytopathological Society. That's a big word. Pacific Division in 2020. And then our other presenter is Dr. Paul Rugman-Jones. He's a project scientist in the Department of Entomology at the University of California, Riverside. He has extensive experience in DNA-based identification and population genetics with the broader context of invasive species management and biological control. He's currently involved in collaborative projects investigating the potential for classical biological control of invasive shothole borers and diamondback moth in California. And so obviously this presentation is more about hope for the future and some of the innovative approaches that are being taken in terms of things like endophytes and then parasitoids.
Thanks, Randall. So yes, Akif and I are going to share this presentation. I'm going to give the bulk of it, and then he will contribute anything at the end if there's anything that I missed here. And then I'll just pass the torch over to Paul. I think that's the plan. And so just thank you for all of those great introductions again. And I just also wanted to say I really appreciated that last discussion, having been all of it, Akif and I and Paul been, involved with this pest since its discovery. I can appreciate the challenges and everything that comes with dealing with it. So here we're going to discuss some of the biocontrol options that we have been investigating, both looking at pathogen side as well as the insect side. So I'm going to talk about the endophytes that we are exploring to control the pathogen side, and then Paul's going to talk about more of the classic biocontrol options to control the invasive shothole borers themselves. And so what do I mean by endophytes? So these are fungi and bacteria that live inside of plant tissues. So just to kind of take a step back, the tree microbiome is basically, it's known to play a role in protecting plants and pathogens and herbivores. And they, just like us, we have a microbiome. The microbiome is just the collective assemblage of fungi and bacteria and even viruses, the communities of microbes that occur on the plant tissues, on and inside the plant tissues. And a component of the microbiome are these endophytes, and these are the fungi and bacteria that live inside of the plant tissues. And these endophytes can be affecting variation in disease outcomes of the hosts. And this is something that we have observed. So for instance, these two pictures of planted sycamore trees were taken in the same location in a disease hotspot in March and November. And the trees with the yellow arrows were infested, but they got better at one point. The trees with the orange arrows were infested and got worse. But some never got infested, as indicated by the white arrow. And, this can simply just mean that, they are susceptible, but not yet infested. But because these are planted trees with similar genetic makeup, we were thinking that another possibility for this variation is the endophytic communities in these non-infested trees may be contributing to whether or not a tree is susceptible or recovering and preventing infection in trees. So to understand how the microbiome can be leveraged for control, I want to just take a little bit of a step back again. We wanted to do this first just to see what the endophytic communities look like and what their role is for different kinds, for the different kinds of microbes that we find, to determine whether wood inhabiting endophytic communities differ among different trees, and if they differ in diseased or non-diseased trees. So each of these plates here, these are culture plates where we've taken wood samples and we've sprinkled sawdust from the wood onto these different culture plates, and they represent plates from wood samples from a different tree, and each of them have differences in the kinds of species and the number of individuals in terms of the microbes that we have found on there. And so there's two important ways to study these microbial communities. They are culture-based approaches where we place plant tissues onto media in Petri plates to see what grows. And there is also metagenomics. That's the most widely adopted, which is metabarcoding here. That uses this kind of high throughput sequencing of DNA from samples to identify the members of the community. There is sampling bias in both for a number of different reasons. And we used both approaches which help kind of mitigate those biases. Today, I'm going to be focusing on the culture-based work because those are the microbes that we can manipulate and potentially leverage and manipulate for control. And so we went out into those plots that I talked about in my earlier talk, and we sampled over 1,200 trees from a subset of those plots to basically screen for endophytes from a whole variety of those of trees. So we collected from sycamores, we collected from willows and cottonwoods and oaks in diseased and non-diseased trees to see what we could find. And we extracted DNA for the metabarcoding work, and we also cultured fungi and bacteria on a subset of those samples, and identified all of those fungi and bacteria to species. And so, not to bog you down with the details, the main thing is just to show this graph is looking at the whole fungal diversity at the family level, with the number of isolates or strains within each on the x-axis. And we just found this huge diversity of fungal endophytes across the host species from a total of 771 strains. And similarly with bacteria, we found a huge diversity of bacteria as well. And they were all represented we found bacteria that were dominated by some species, but we found that they were all represented by different groups as well. And so we did an analysis of everything that we cultured to see if the endophyte communities among disease and non-disease trees. And we found this really interesting pattern where the microbial communities in non-disease individuals had different compositions than their neighbors had that had been attacked. So the black dots are trees that are not diseased. The pink dots are trees that were diseased. And so things that are closer together are more similar. Things that are further apart are more different from one another. So this is the culture-based results that we got. And they raised some interesting questions. You know, some of the questions are, once the trees are attacked, do the microbiomes change? Or do pre-existing differences in the microbiomes of these trees protect them or make others more susceptible to attack? So there are some interesting questions that we're looking at, but it does raise the possibility that there's some microbes in there that are actively protecting trees from attack by Fusarium as well. So in our preliminary screening, we tested these 60 fungal and 40 bacterial species for inhibition of the Fusarium pathogen in a culture experiment. And what we did was we placed a mycelial plug of Fusarium in the middle of an agar media plate. And then we put different endophytes on the edge of those plates of the margin, and we allowed them to grow. The plate on the left is a Fusarium culture without endophytes, and the plate on the right is a treatment where isolates that cause inhibition are indicated by the red arrows, which exhibit what we call this zone of inhibition, where basically there's these antibiotics that are diffusing through the media, and they're preventing the fungus from growing further to the edge of the plate. And the isolate here that's indicated by the blue arrow could not prevent the fungal mycelia from growing to the edge of the plate. So it didn't cause any inhibition. But we saw this range of interactions between the fungi and pathogen, including antibiotic inhibition to competition to coexistence. So we know the behavior of some of these species that might inform our analysis for a richer sequencing data set as well. And taking this a step further, we took any bacteria that exhibited antibiotic inhibition in vitro, so in those plates, and we inoculated them into avocado and sycamore seedlings. We allowed them to establish. So this plate here, this is from, you know, those woody pieces on the right showing confirmation of their establishment. And once they were established, we inoculated them with Fusarium to see if the Fusarium could colonize those seedlings that had the endophytes established. And 3 months later, we tried to re-isolate the Fusarium from the woody tissue. And this graph is basically showing that the proportion of trees that we re-isolated Fusarium from seedlings. And what this gives us is an indication that when seedlings were co-inoculated with these bacterial endophytes and Fusarium, they inhibited the establishment of the pathogen. And so this is important because the beetles can only survive if they can eat this fungus. And remember, the fungus itself is a pathogen as well. So if the microbial communities are interfering with the fungal growth, it protects the individual plant and further reduces the spread of disease as well. And so where we see this having the most immediate application is in restoration settings, where you can manage the microbiomes of organisms before they are planted into the wild. So we're working on doing different inoculation techniques or different ways to introduce these endophytes into plant material to deliver them into cuttings on a large scale for that particular purpose, and trying to find out how to introduce them for nursery and restoration applications. And one other thing I wanted to talk about is because there can be changes in the microbiome over time, you know, things can change seasonally and everything. We're also kind of exploring with our colleague at UC Riverside, Jason Stach, how these things can maybe even change seasonally too, if there's any shifts in those communities over time as well, and how that might influence how we manage microbiomes in, for example, restoration practices. So that's all I have to say. And I'd like to turn it over to my colleague, Akif Escalin, to see if he has anything else to add.
Thank you very much, Shannon. You have done an awesome job explaining what we have done for the biocontrol. So I just want to mention that the reason that we started focusing on beneficial endophytic microorganism is that I also have seen a lot of questions in this chat that we are just looking for how we can control this pest and disease complex. The challenging part of controlling this pest is that there are a few reasons. One, we are dealing with more than one pest, including beetle, more than one beetle species, and their associated fungal pathogens. To manage or control or find one single bullet kind of thing is very difficult because you are dealing with two different pathogens or two different entities. That's one. The second is that the beetle and their fungi lives most of their life within the plant, within the woody tissues, where you cannot deliver any chemical easily. Especially people have been asking a lot of questions, how we can apply the contact effect pesticide, including fungicide, insecticide, or all kinds of things. And also there are very limited number of the systemic pesticide that could be delivered through the xylem, where you can have the control of it. Even if you have it, the protection is going to be the shorter time because plant is active. Those pesticides going to be disappearing within the short time. Because of all these reasons, we found out that the only sustainable way of managing this pest could be either biocontrol using the endophytic beneficial microorganism that Shannon mentioned, or finding an effective natural enemies that Paul is going to talk about it. That's all I wanted to say. So we are dealing with very complex issues, the difficult that brought us to these two chapters that we would like to do. Thank you.
Okay, so I just want this morning just to run through some of the work that we've been doing at UC Riverside on potentially developing or looking at the potential for developing a biological control program for these invasive shothole borers. So this is work that was originally started by Richard Stouthammer, someone that I've worked with for the last 20 plus years. He retired 2 years ago, and I've sort of taken on the role of leading this effort now here at UC Riverside. So for anyone who doesn't know, biological control, classical biological control involves the introduction of a natural enemy from the native range of the pest, this pest, in this case, Southeast Asia. And that would then be released under certain, after undergoing various testing, it would be released in California and hopefully affect control of these shothole borer populations. And for almost 7 years now, we've been working with a group in Taiwan. We chose Taiwan because some of our earlier genetic work told us that both of the species that have invaded California are both present in Taiwan, so Kuroshio shothole borer and Polyphagus shothole borer. And following some quite extensive field survey work over there, some monitoring work, and just simply collecting infested wood, shipping that back to UCR under permit. We've actually imported several natural enemies now to the US, which were originally coming to the quarantine facility here at UCR. And more recently have been going to the USDA facility in Massachusetts. So this is a quick overview of the process. So in Taiwan itself, we have people on the ground. Fang Ling, Yi Chang is my current postdoc. Ricky Lara, now at the CDFA, was a previous postdoc based in Taiwan. They visit infested sites, cut wood. This is, I believe, Akif vandalizing an avocado tree in Danai, in Taiwan. So we cut infested wood, we package that up, and we ship it under very strict permitting conditions back to the insectary, like I said, at UCR or now Buzzards Bay, Massachusetts. And we wait to see what emerges from that wood. And so far that work has yielded 3 potential candidates for biological control. All 3 of these are parasitoids. They're all brand new to science. There is very little work done on natural enemies of ambrosia beetles, to the point that when we first started looking at this, people who work on ambrosia beetles simply told us there are none. Well, there's 3 examples here that suggest they're wrong. So all 3 of these have been imported to the US. Unfortunately, 2 of those, at this point in time, we've struggled to establish colonies of those in the quarantine facilities, and we require those colonies simply so that we can then proceed with the sort of testing requirements required of any development of any biological control program. And so those 2, for now at least, have been abandoned. But the 3rd one, this species in the genus Phymasticus, we've had some success with. Surprise! So this species is actually was actually the least abundant in the wood that we obtained from Taiwan, but it's the one that we're best able to rear, and we have very good evidence of that it's an effective parasitoid in Taiwan. And I'll show you a slide in just a second that shows some of the evidence for that parasitism. And importantly, we've been able to establish those laboratory populations. So this is this is our proof that it's good in the field. So this Phymasticus is an endoparasitoid. A female wasp finds a beetle. You may have noticed in the work that you've done yourselves that the beetles have a habit of hanging out in the entrance of their gallery. They tend to leave their butt exposed. We think this is simply a protective behavior to stop things from entering the gallery, but also leaves them somewhat vulnerable. And one of the things they're vulnerable to is this wasp, which comes along. It lays 2 eggs into each one of these beetles, a male and a female. Those eggs hatch, the larvae develop within the beetle, and then eventually when they turn into adults, they chew their way out of the abdomen of that beetle and move on to look for their next victim. So that looks all quite promising so far, our work in Taiwan. We're currently at the point where we're about to start doing some non-target testing on that species of Phymasticus. But we were also thinking about this, and Taiwan is quite honestly is unlikely to be representative of the full range of natural enemies that might be associated with this beetle complex. So this is just a map showing the approximate native range of the whole Eualacia fornicatus species complex. And you can see that Taiwan is just a very small part of that. And so in summer last year, we extended our search to Thailand, in particular to northern Thailand, where we know that the prolific shothole borer is. This is just a map showing you some of the sites that we're working at. So we're collaborating with people at Chiang Mai University, and we're working in mountainous areas in the province of Chiang Mai and neighboring provinces of Mae Hong Son, Lan Phun, and we're hoping next year to go to Chiang Rai as well. Chiang Mai itself is a very sort of cosmopolitan city area, but the surrounding areas are very sort of remote and isolated. It's just a quick summary of what we're doing in Thailand. So this is a group we're working with. This is my collaborator, Dr. Bajari Chitong, or Bom as she goes by. And this is her team of assistants. One of the biggest problems with this work is actually not problem, but the biggest challenges in this kind of work is actually establishing a contact in the first place. So Bom is actually, he's not interested in per se in ambrosia beetles. She doesn't work on ambrosia beetles. She actually works on honeybees and stingless bees. But importantly, she has really good contacts in these mountainous areas, are able to then grant us access to their property. I think the most exciting part on this slide is this photograph here. You remember those exit holes that I just showed you? This is an exit hole produced by exactly the same wasp that we have in Taiwan, the Phimasticus. So we're really excited about that. At this stage, we haven't proceeded to the point where we will be importing this material, but that is in the pipeline. So again, this is my collaborator. If you're interested in looking up some of the work that we've been doing, she features it on her Facebook page, although the majority of that is on her own work on these. This is an extension leaflet that with the help of BOM, we produced for distribution to local landowners and farmers in the Chiang Mai region. And it really is, I can't really overstate this point, but without BOM's help, we wouldn't be able to do this work. So her contacts basically allow us access to all of that land. And that land is all sort of governed by two projects, the Royal Project and the Highland Research and Development Institute, both of which provide funding to the hill tribes in northern Thailand. And effectively their program seeks to replace what was once an area where they produced all the opium. And they replaced that with a range of crops, one of which is avocado. So there is a lot of avocado grown in that area. And of course, we're concentrating on surveying the avocado in those areas. Plus, there's quite a bit of castor bean in that area as well. So we do sort of check castor bean as well. And so it all looks promising, but as of March of this year, in response to budget cuts, federal funding for ISHB work has been reduced. USDA-APHIS no longer consider it a prime pest. And so we're having to, so in the past, we've relied on appropriated funds for this work. So there was a certain amount of funding every year that was available specifically for shothole borer work and our work in terms of foreign exploration and biological control. That's gone. But we are currently exploring other funding sources, essentially to complete our non-target testing of that Thymasticus species, the Buzzards Bay in Massachusetts. Also to continue the exploration in Thailand. But we want to go further than that because one of the other, another area that we're interested in is Okinawa. So again, our early work showed that both Polyphagous and Kuroshio shottle borer were present in Okinawa. Okinawa, there's a recent study from Japan reporting Polyphagous shottle borer as a serious pest of mango in Okinawa. And so we're keen to go to Okinawa and survey their mango orchards and see if there is anything there that might be of any interest. With that, I'll just thank all the people who've contributed to this project. If people have questions, then I'm happy to answer them now. If they have questions in the future, then please feel free to email me. And that's it.
Very good. Thank you. Thank you, Paul, Latif, Shannon. Excellent.
So, is there any chance the pest can be extirpated in Northern California, or is it already too widespread?
I thought Akif actually answered that question already, but do you want to take that one, Akif? Is it possible to eradicate it up there?
Oh, eradication? I think I answered that question as a response. In California, I don't think eradication of this pest is possible anymore. That's what I said, that we have to learn how to live with this beetle by monitoring, applying the cultural practices, including integrated pest management and other management strategies that we all talk about it during this conference.
And that is true in San Jose as well as the other places where we have this pest.
Yeah, one of the reasons is that the one single beetle, if survives, it can start a new colony without even needing male component of it. It's like that complex of the beetle, and then they are carrying their fungus. And then just to mention that the genetic studies that we have collaborated in the past, that this beetle and their associated fungi has been around more than 2 million years. Imagine if this beetle and complex has been survived that long during the time, so they have a very protected lifestyle, which we have been talking about.
There's a question about, with concern about spreading with a lot of green waste and regulation of green waste in the state now, spreading through woody materials, and wants to know if there are any efforts going on statewide to maybe regulate or work with some of these services that share, or unregulated, that share woody materials that may not be properly processed.
I wonder if Shannon or maybe Bea could come back on and address that question.
I can add some after you guys.
Well, here's Bea's.
But no, go ahead, Akif, if you.
Okay, let me start, and then you guys go ahead. The problem with that, yes, the state may regulate and everything, but I don't know if that would work, because beetle has a wide host range that we all talk about it. If a single infested branches in someone's backyard could be cut off without knowing it, put into the green bin, and that could be moved longer distances. Because of that, each city should be considered putting trap or monitoring the beetle infestation in their green waste managing areas. In the past, we have identified some beetles nearby the green waste has been managed in California. So that could be something that can be done in that area. Otherwise, I don't think we can regulate people not to move infested plants because there are many different the host that cannot be identified. Some people may distribute the plant material without knowing it because in their backyard. That's my thought.
A couple of, unless Bea or Shannon, do you want to add to that?
The one thing that I could add is that from the working group on invasive tree pests, the thing that we have been doing is reaching out to this green waste management facilities with information. So there's no regulatory power here. We can't say, oh, you're canceled because you're not following this or that recommendation, but we can provide information and recommendation on how to do things to prevent the spread. So among those things is, for example, not letting the wood do not stage the wood somewhere to be processed later. So try to get the wood processed as soon as possible. And that's something that usually the green waste management facilities don't do. They tend to process it fairly quickly. And in fact, I know of some places that if the wood is flagged as infested, they will charge a little bit extra, but they just put that load to the front of the line to be processed earlier. But something that municipalities can do to prevent the spread is to avoid having staging areas, which is some cities would just cut all the green waste, put it in a place until they get a big enough pile or something to send to be composted or something. So those areas where the infested wood stays for a while, that's where the beetles also can come out and infest the surrounding areas. So that is something that, even though there's no regulation, that is a really good thing that municipalities can tackle, the not having those staging areas.
I also saw that Sara Davis had her hand up. Sara, if you want to come back on, you should be able to talk as well.
Yeah, I mean, we can regulate as much as we want, but that's never reality. People move stuff. I don't know why they do it, but they can be on a 4-wheel drive Jeep trail in the middle of nowhere and get to the top of a mountain, and someone has dumped their old washing machine.
Mm-hmm.
It's really hard to control the movement of things. I know in San Jose we have the most bizarre green waste system I've ever seen, where you basically take all of your green waste and you just dump it in the street, and then this weird little tractor thing comes by and picks it all up. There's no bin, so we have I can't conceive of a way that we can control the movement of infested wood. It's just there's too many people involved in the system.
We have the same issue with firewood as well. It's not possible to regulate it. Certainly would cost a ton of money to get regulations in place, and then following that, the enforcement. There's just not a budgetary support for that. So, most of this is a matter of providing information and trying to get the message out, not to move firewood, not to move green waste.
Julie, there's a couple of questions there about native predators.
Yes, take it away.
Okay, so to answer those two questions, I am actually currently writing a grant proposal to actually do a survey for that kind of for any indigenous natural enemies that might have switched on to shothole borer. We've seen this is based in large part to what we saw in the Tijuana River Valley, where in 2015 you couldn't find a tree that didn't have a shothole borer, a Kuroshio shothole borer. You go down there now, and I'll challenge you to find a tree that does have a shothole borer in it. But they are there, and at one point during COVID, we actually collected some wood down in Tijuana Valley with the help of John Boland, who knows where those infested trees are. And we brought that back to the lab, and we did actually have one parasitoid emerge from that. At the time, we were quite excited about it. It was a Neocalisota species, and it's one that is associated with walnut twig beetle, which is a similar native pest here in California. We haven't recovered that again. We've done a little bit of a preliminary work collecting infested wood and seeing what comes out of it. But I'm currently seeking funding to do a sort of rigorous survey of some of the sites, particularly here in Southern California, that were formerly infested and not. They now still support an infestation, but perhaps not quite as heavy as previously. And we'll see. I mean, it's something we're thinking about, and obviously, if we can find an indigenous natural enemy, then it would be a heck of a lot cheaper than a heck of a lot easier than seeking all of the negotiating the red tape involved with bringing in a biological control agent and releasing it from a foreign country.
Thank you, Paul. Anyone have to add to the biocontrol research question? There's a question wondering what kind of outreach efforts are going on in Northern California considering the Santa Clara County infestations, specifically to adjoining counties, agencies, academia, other institutions.
Unfortunately, Igor got off the call. He had to go to another webinar where he's presenting, but he has been meeting with a lot of organizations, reaching out in the Northern California area. And we've done various trainings in San Jose and beyond, and there will be a lot more of those to come, certainly.
Well, and Tanner Marr, our regional CAL FIRE person, who's been working on the incident action plan. There's a small group of us, and myself, Igor, Tanner, and a couple others have been trying to identify the strategic people that we need to have conversations with to get them on board so that we can kind of collectively act regionally. So the entire Bay Area say, hey, let's work together, because pests don't just stay in one jurisdiction, they move around. So behind the scenes, we've kind of been working on finding the right people to bring together in a collaboration.
And that does it for our open questions.
Again, thank you to all of our presenters today. This, I think, was a successful program. I've gotten some comments about things people would like to see at future events like this, and we'll certainly take those into account when we plan our next session. Thank you all.